Hello. 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 Right, sir. Right.
Good morning, everyone.
Yeah, good morning, madam. Good morning,
Kindly,
sir. Good morning,
Sir,
good morning, good
good
morning.
morning, sir.
Yes, sir. Yes, sir. Good morning, sir. Welcome to Nehru College of Pharmacy, sir. Pharmacy, sir.
Thank you, sir.
I request everyone to kindly mute your mic and off your cam. Very good morning to all. Welcome everyone to the third day of International E-Faculty Development Program organized by Nehru College of Pharmacy. Today we have Professor Dr. K. Ishwar Kumar sir for today's session, morning session. Professor Dr. K. Ishwar Kumar, M.Pharm PhD, obtained B.Pharm degree and M.Pharm and PhD from Andhra University, Andhra Pradesh, and awarded Best Thesis for PhD in the year 2006. He is having 22 years of experience in teaching and research. Dr. Professor K. Ishwar Kumar, elected as the Vice President, Association of Pharmaceutical Teachers of India, Andhra Pradesh in 2014 to 17. Elected as a Vice President, Association of Pharmaceutical Teachers of India, Andhra Pradesh branch. Joint Secretary, Indian Pharmaceutical Association, Visakhapatnam. Co-convener, Andhra University Research Forum. Secretary and Treasurer, Indian Pharmaceutical Association, Visakhapatnam. Sir is an advisory board member of Suraksha Pharma Private Limited. Sir received many prestigious awards like awarded Best Paper for in the 10th Annual Conference of IPS Orissa. Awarded Best Paper in National Seminar held at Department of Pharmaceutical Sciences, Andhra University. Professor Desharaju Venkatrao Endowment Prize for the Best PhD Thesis in Pharmaceutical Science for the year 2006 by Andhra University. Best Research Award in 2017 by Andhra University, Visakhapatnam. Best Teaching Pharmacist Award 2017 and National Award by Simanta Drug Dealers Association and AP Private College Management Association. Professor P.P. Suryakumari Award for the Best Paper published in Indian National Journals in Golden Jubilee Celebration of Indian Pharmacological Society held at Mumbai. Best Research Supervisor Award from DKIRF International Society held at Chennai during 13 June 2019. So was the resource person at MHIRT program on MOU between Andhra University and Alcorn State University, USA. External subject expert for B.Pharm syllabus division of Rajiv Gandhi University of Health Sciences, Bangalore. Observer for ICT CMAT examination, ICT GPAT examination. Is a PhD thesis evaluator for many universities. Is professional member in many associations like Pharmaceutical Pharmacological Society, etc. He graded 27 PhD students and awarded, and 4 are under PhD's adjudication. Also, he graded 44 M.Pharm students under Professor Dr. Ishwar Kumar sir. He published his research work in many national and international reputed journals. He is a visiting scientist in Department of Physiology, University of Malaya, Kuala Lumpur, Malaysia. I welcome this eminent person for today's session. Welcome you, sir.
Thank you. So, good morning, Dr. Sudhahar, Principal, Nehru College of Pharmacy. Sir, is it audible? Let me check once. Somebody please confirm.
Yes, sir. Yes, sir. Yes, sir. Is it audible, sir?
Sir, okay. Thank you, sir. Thank you. So, and the management of Nehru College of Pharmacy, the organizers of the faculty development program, all the faculty, and the participants of this faculty development program. A very good morning to all of you. Sir, can you please check my presentation whether it is visible or not? Is my presentation visible, sir?
Yes, sir. Yes, sir. It's coming,
Right,
sir.
right, sir. Right, sir.
Please.
Yeah, thank you.
Yeah, kindly proceed, sir.
Right, sir. Thank you. So, good morning to all of you once again. So, today I'm going to present my topic on trends in pharmacology research. So, the pharmacology research, actually, my aim of presenting this topic is to make aware of the research in pharmacology, particularly the budding pharmacists. So my focus mainly on the budding researchers, particularly the informal students and those who have joined the research PhD programs recently, and also the teachers, young teachers. So please, the remaining, please bear with me. So I'm going to give some basics which may be known to many the senior faculty. So for the benefit of the students, the young faculty, and the research scholars and the informal students, those who are in research, for those people, I would like to give some information regarding the pharmacology research, or what the trends are there in pharmacology research. Okay. So let me explain. So about the pharmacology. Pharmacology is a subject. So it's so important for any medical professional, not only pharmacy. So pharmacology is there in all medical professionals, starting from the doctors, pharmacists, nursing, and other paramedicals. So they read the subject pharmacology. So as we know, the pharmacology majorly having the divisions like pharmacokinetics, pharmacodynamics, pharmacotherapeutics, pharmacy. So here what I want to focus mainly, what I want to impress you upon is the importance of pharmacokinetics. So those who are in the field of pharmacy must be aware of the pharmacokinetics. So all other medical professionals, healthcare professionals, they mainly focus on the other divisions like pharmacodynamics, pharmacotherapeutics, and other divisions of the pharmacology. But as being a pharmacist, we should know, majorly we should focus on the pharmacokinetics, what I believe. So I need not explain much about these divisions, what they tell about. Simply, pharmacodynamics means what after taking a drug into the body, when it enters into the body, what it is making, what kind of changes it is making in the physiology of our body systems. So reading about those changes is a pharmacodynamics, simply I can say. Whereas pharmacokinetics means it's about the fate of the drug, how the fate of the drug changes in the body after its administration into the body. So pharmacokinetics majorly we can divide them into absorption, distribution, metabolism, and excretion. Absorption, as we know, it is the transfer of drug molecule from site of administration into the systemic circulation. It is called as the absorption. So absorption majorly involves there is a crossing of the biological barriers. So ultimately we require the concentrations of the drug in the systemic circulation, that is in the blood circulation. So wherever there is a barrier, they cross the barrier and enters into the systemic circulation. So in case of IV administration, we consider it is as 100% available, 100% absorbed, and distribution, the drug, how much a drug is moved to various body tissues and organs depending upon the flow of blood through the tissues. So distribution means the drug has to be distributed uniformly throughout the body. It has to reach to each and every tissue then only it can reach to its site of action, then only it can produce its effects. Then metabolism and excretion together, we call it as elimination. So what are the drugs we inject into the body, they are considered as foreign to the body, foreign substances. So our body immunomechanisms, they try to eliminate the drugs from the body. In the process of elimination, the drug gets metabolized. Metabolism means the drug is broken down into smaller molecules and gets excreted by excretory organs. So if you see the phases of drug action, if you take the blood samples at different time intervals after drug administration into the body, if you consider if it is oral administration, there is some kind of absorption and distribution, and there is a metabolism and excretion. The initial part, the curve shows the absorption and distribution, and the later part, the declining part, that indicates the excretion, the elimination of the drug from the body, that include metabolism and its excretion. So distribution, so mainly remember the drug after enter into the systemic circulation, it should be free in the systemic circulation. When the free drug, when it is in the free form, then only it can be available for its effects. It can reach to the site of action, it can produce its effects. So sometimes some drugs are bound to the proteins that are present in the systemic circulation. Okay. So some drug may be stored in the reservoirs like tissues and even in the fat. So such drug may not be available for its producing its effects, even for its metabolism. When it is in the free form only, that can produce its effects and it can get excreted. So there is a called as first pass metabolism. Certain drugs undergo first pass metabolism. First pass metabolism is nothing but the drug before reaching its systemic circulation, it gets some of it gets metabolized in the portal circulation by the liver, because liver is the major site of drug metabolism. So most of the drugs are metabolized in the liver. So some drugs they undergo first pass metabolism. So see this figure, this indicates the therapeutic effect. We can know therapeutic window. This is the graph indicates the time versus blood concentration curve. So this is oral administration. You can see the onset of action. See, this is the like a parabola. So this is called as the minimum effective concentration, MEC, and there is called as MTC, minimum toxic concentration. So any drug to produce its therapeutic effect, it has to cross that minimum effective concentration. Remember, that means it should achieve certain concentration in the blood, then only it can reach to its site of action and it can produce its effect. That concentration is called as minimum effective concentration. And at the same time, all the drugs are chemicals. They are toxic at some point at certain concentration. So every drug will have its own toxicity levels. So this is a minimum toxic concentration. So any drug, after crossing its minimum effective concentration, it should remain below the minimum toxic concentration to produce its therapeutic effect. So the gap between minimum effective concentration and minimum toxic concentration is called as therapeutic window or therapeutic range or therapeutic margin or margin of safety. So any drug which have the concentration in between this minimum effective concentration and minimum toxic concentration, that can produce its effects. So there are drug targets. So drugs, there are targets for drugs. These targets are like receptors, enzymes, hormones and hormonal factors. Some target directly on the DNA, some on nuclear receptors, some they target the ion channels, some act by unknown mechanisms. So most of the drugs, they target the receptors. So if you see the statistics, I got this from Goodman and Gilman textbook. So the receptors, for about 45% of drugs they target the receptors, about 28% they target the enzymes, about 11% they target the hormone and hormonal factors, and 2% DNA, uh, and 2% nuclear receptors, 5% ion channels, and some 7% by unknown mechanisms. So these are the major targets for drug effects: receptors, ion channels, enzymes, and carrier molecules. I would like to give some examples for these receptor targets. For example, there are many kinds of receptors are present. For example, acetylcholine, there are two kinds of receptors: muscarinic receptors and nicotinic receptors. Similarly, adrenergic receptors, receptors for 5-hydroxytryptamine, receptors for dopamine, receptors for insulin, receptors for estrogen, like that. There are agonists and antagonists at these receptors. For example, nicotinic receptors, acetylcholine receptors, nicotine is the agonist and D-tubercularine is the antagonist. So like that, ion channels, there are different kind of ion channels are present they are located on the cell surface, on the cell membrane, like voltage-gated sodium channels, in the renal tubular sodium channels, voltage-gated calcium channels, ATP-sensitive potassium channels, chloride channels present in the brain, and glutamate-gated cation channels. Like this, there are different ion channels. They when the drug occupies these channels, the drug allows the ions to flow into the channels, and that makes the cellular changes. That's how some of the drugs, like local anesthetics, like anti-arrhythmic drug, and some of the drugs like picrotoxin, all these drugs, they by influencing, by modifying the ion channels, they produce their effects. And coming to the enzymes, so there are enzymes in our body. The enzymes, they are involved in many metabolic processes in our body. For example, cyclooxygenase, we call it as COX enzyme, which is involved in the production of prostaglandins, we know. So the prostaglandins is inhibited, prostaglandin synthesis is inhibited by the non-steroidal anti-inflammatory drugs like aspirin. So that is by inhibiting the cyclooxygenase enzyme, it inhibits the synthesis of prostaglandins. Like that, there are enzymes which are targets for many drugs. Another example is ACE, angiotensin converting enzyme. So the angiotensin converting enzyme inhibitors like captopril, lisinopril, ramipril, all these, they by acting on ACE, they produce anti-hypertensive effects. They reduce the blood pressure. Similarly, HMG-CoA reductase, monoamine oxidase, dihydrofolate reductase. Like this, there are enzyme targets for drug act. And there are some carrier molecules which are also targets for drugs, like choline carrier, noradrenaline uptake, vesicular noradrenaline uptake, sodium potassium chloride co-transport, and we know the proton pump and sodium potassium pump. Like this, there are targets for drugs. So how the drugs produce actions? So this is directly by acting on the receptors. They modify the entry of ions into the intracellular, and they cause activation of the conductance, like sodium ion channel facilitators, sodium channel blockers. They act on by similar mechanism. Similarly, there are like we call them as G protein coupled receptors. They activate the cell signaling by generating the second messenger. So, similarly, some drugs they modify the receptors, like tyrosine kinase receptors, phosphorylation of tyrosine kinase receptors. That is, they activate the cell signal. Tell me, sir.
Sir, extremely sorry for the interruption, sir. And screen is blinking, sir. Not continuity is not there, so could not able to the view properly, sir. Kindly, there is a blinking is there.
Once again, shall I share the screen?
Sir. Just you can disconnect it again, sir.
I'm sharing once again.
Yes, sir. Yes, sir.
Sir, is it okay now?
Okay, sir.
Sir, sir, actually, so these, I'm giving some basics about the drug actions and the drug receptors before going into the actual research topics, how we go into the research. So this is
Thank
about
you, sir.
the
Thank you,
source.
sir.
Sir, okay, sir. Yes. So this is about the sources of drugs. We know the drugs are obtained from many sources like plant sources, animal sources, mineral sources, microbial sources, and most of the drugs are from synthetic sources, semi-synthetic sources, and some drugs are obtained by uh, like recombinant DNA technology. So this is the process of drug development. You might be knowing. Yesterday, I think you have got some very good lecture from other speaker on drug development. So there are stages in the drug development like target selection. First of all, whenever we go for a new drug for evaluation of new drug for any pharmacological activity, if the drug or a compound which is completely unknown, so such drugs need to be means there are steps involved in the development of those drugs into therapeutic molecules. So before going into any drug development, we need to know the target, means where to target, what is our requirement. So that means, for example, for diabetes, if it is a diabetes, in case of diabetes, what to target in the diabetes? What means what area means like directly want to target the insulin secretion or the mechanisms which are responsible for insulin secretion or the molecular mechanisms which involve in the generation of pancreatic beta cells, like that we have to choose that target selection first before going for the drug development. So where to target, like cellular or genetic targets, which may be genomic level or proteomics or bioinformatics, basic using all these the technology, we can target the selection. First of all, target selection, then lead discovery. The lead discovery may be, as I told you, there are many sources for drugs, like animal sources, plant sources, microbial sources, synthetic sources. Like this, the first of all, we have to identify, we have to discover the lead by either by synthesizing or by isolation or by combinatorial chemistry. So, or by using the technology like high throughput screening. So, like this, the lead discovery is the second step. Then comes to the medicinal chemistry. So we can using there are different softwares that are available nowadays. We can check whether our lead molecule can target our whatever target we have fixed. So that we can check in vitro without means before going into the actual studies by using there is a library of compounds and we can do some SAR studies for that molecule, and by using some in silico screening techniques. So all these processes we can use to identify whether the lead molecule, whatever we have synthesized or isolated, which can target our selected target or not, we can know before going into the actual studies. Then we can conduct some in vitro studies. So actually the pharmacology starts from here. So in vitro and then in vivo, then it goes to the clinical trials. So in vitro studies, before subjecting the molecules into the animals, so they can be tested for their safety, for their affinity, not for safety. For their affinity, we can do some in vitro studies, and we can know the affinity, whether they are having that affinity towards that particular target of a particular receptor or enzyme or whatever it may be, and we can fix the disease models, and by taking the disease model, we can in vitro also we can fix some disease models like a cell line studies. So by using cell line studies also, whether that particular target molecule means the lead molecule is actually producing effect or not can be tested, and we can know to some extent the mechanism how it is going to produce its effect. So like this, by using in vitro studies, we can know the affinity of the drug molecule towards the selected target. Then the next step is in vivo studies. In vivo studies normally conducted in animal models before going into human trials, human before subjecting into humans. The in vivo studies, first of all, the safety studies are conducted, and at the same time, they have to be the compounds have to be tested for their efficacy. Safety and efficacy studies are conducted by in vivo in animal models. Then once
Sir, again, sir, excuse me, sir. Again, the same issues are there, sir. Blinking is there, sir.
no, sir, no, for me it is okay.
Oh. Now it is okay, sir.
Okay.
As such.
Okay. Okay.
Yeah.
Right, sir. So there are means our pharmacology starts from in vitro studies and in vivo animal studies. So that we call them as preclinical studies. Before going into the clinical studies, they have to be subjected for tested for their efficacy and safety first. The preclinical studies can be divided into in vitro or in vivo. So in vitro studies, again, in vivo studies, they can be pharmacological or toxicological studies. So pharmacological studies to determine the efficacy of the compound, and the toxicological studies to determine the safety. Remember, always a compound should have efficacy, at the same time it should be safe. So from that efficacy and safety studies only, we can calculate the dose to be administered for its activity. So we can determine the starting dose from efficacy and safety studies. Remember, we should maintain the balance always between the safety and efficacy. So the true balance is always required. If the compound is more efficacious and which is not safe, that means which is having some toxicity or producing some more adverse effects, and adverse effects are severe compared to its potency, then that drug cannot be subjected for human studies. Similarly,
sir, sorry, sir. Sir, screen is not showing, sir. Sir, you please click present screen, the present screen and whole screen, sir. Entire screen. Select the entire
screen only. I'm presenting entire screen only, sir.
Okay, sir. Now the screen is not showing, sir.
So now you see, sir?
No, sir. Again, the fluctuations are there.
మనం రికార్డ్ చేయాల్సి వస్తుంది తర్వాత నెక్స్ట్ వచ్చేసి
sir, once again I'm presenting. Sir, is it okay now? Hello.
Can increase the percentage, sir, in this only.
Yeah.
That would be okay, I think. Sir.
Is it okay now?
Okay, sir. Now it is okay. Again, no, sir. Again, the same problem, sir.
Again, same problem. Okay, I'll do one thing. Presentation mode I have to remove, I think.
Yes.
It is not going in the presentation mode.
No, text is not going, sir. No. Probably 80% is. Okay, sir. Now.
Okay, no, sir?
Yeah, that's fine.
Okay. So the safety and efficacy, always we have to maintain the balance. Compounds should be safe, at the same time it should have the required efficacy, then only it can be tested for in vivo studies. Is it okay, sir?
Yeah.
Okay. So for any new compound, any new substance, for example, any herbal compound, we what we don't know the pharmacological activity because so which is not claimed for any pharmacological activity, which need to be evaluated for its pharmacological activity. First of all, so it is evaluated for its adverse pharmacodynamic effects. Hello? 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 Sir, is it okay now?
It's clear, sir.
It's clear, no?
Yes, sir.
Yes.
Okay. Okay. When we go for evaluation of a new compound, so first of all, the safety pharmacology, there are like a core battery of studies we need to conduct. When the compound is completely unexplored, when we know when we are unaware of its pharmacological effects, then the first activities that need to be tested are its effects on the central nervous system, the cardiovascular system, on the respiratory system. Then the follow-up studies for safety pharmacology, the other systems. So this is what we call them as a functional observational battery of tests. So in the central nervous system, the core battery of studies are like motor activity, what is the effects on the motor activity, then the behavioral changes, coordination, sensory and motor reflex response, and the body temperature. So these are all the tests that have to be conducted. I think some of you might be knowing about the blind screening of the drugs when we don't know the compounds nature. Before knowing its chemical structure, also we can screen such compounds for their pharmacological activities. These are all the general tests that have to be conducted before subjecting them for a particular activity. So this is for general screening. We call them in the blind screening. So this apart from these core battery studies, the follow-up studies need to be conducted for effects on the learning and memory, the ligand-specific binding, neurochemistry, visual and auditory examination. These are all the tests that have to be conducted for safety pharmacology. The functional observational battery of tests for like neurotoxicological studies and neuropathophysiological investigations have to be conducted. This is called the Irwin's test. So in the modified Irwin's test, all these neurotoxicological and neuropathological investigations are incorporated. So other established techniques, by using these techniques also we can screen such compounds. For example, using the rotarod instrument for muscle relaxant activity, we can know the compound is having any muscle relaxant activity using rotarod instrument. And for knowing its analgesic activity, we can know we can use heart rate test, tail flick method, top pressure method. Like that, there are different kind of methods are available to evaluate the analgesic activity. Then There are photoelectric beam interruption techniques to know the antidepressant activity. We know that active photometers, most of the students are aware of all these instruments because all these instruments are mandatory according to Pharmacy Council of India. You might be having all these instruments. Some of you might have worked on that also. So this generally we can know the antidepressant activity by photoelectric beam interruption technique using a photoactometer. Then passive avoidance test using a pole climbing apparatus, and the pentylene tetrathol induced seizures from that we can know the any convergent anti-convergent activity, and electroencephalogram EEG we can record the EEG and we can know any central effects of the compound. And there are like many instruments are nowadays available to know directly. We can visualize the changes that are taking place in the central nervous system, also in the peripheral organs. So in vitro studies. So apart from in vivo studies, there we can also conduct some in vitro studies like we can know the potency of the compound by activity assays, bioassays, and we can also conduct some toxicity assays to know the toxicity by in vitro studies. That is by using either isolated tissues or organs like a skeletal muscle, smooth muscle, or aorta of the heart or entire heart, and chemical assays also we can do to know the potency, and we can use the cell culture studies to know the potency of a unknown compound, new compound. So after knowing its pharmacological activities, the drugs has to be tested for its toxicity. So toxicology is defined as a study of poisons and concerned with the adverse effects of the xenobiotics. So toxicity means it is not only the toxicity at the toxic dose levels, and also the adverse effects which results with the therapeutic doses of the drug. So the toxicological screening is important for the development of new drugs and for the extension of this therapeutic potential for existing molecules. These toxic tests are mostly used to examine specific adverse effects or specific endpoints such as cancer, cardiotoxicity, or like other toxicities, irritation on the skin and eye. So according to Paracelsus, the father of toxicology, all the substances are poisons. There is none which is not a poison. The right dose differentiates a poison and a remedy. So as being a pharmacist, we must know the right dose which differentiates poison from a remedy. So the dose which produces the therapeutic effect without producing any adverse effects, without producing any toxicity, that's what the duty of a pharmacist. And we need to know the ethical considerations before going into any studies in animals or on the tissues or cell lines. These studies, before conducting these studies, should be approved by our institutional animal ethical committee, and the institution or the lab should be recognized by CPCSA, Government of India. The objectives of toxicity studies are to know the benefit and risk ratio can be calculated from that, and also we can also calculate the therapeutic index. So therapeutic index is the ratio of maximum tolerated dose to minimum curative dose. So if this ratio is smaller, the drug is having, we can say the drug is having better safe. So that's how we can know the therapeutic dose, and the safe dose can be calculated using toxicity studies. Also, we can determine the LD50. From that, we can know, we can calculate the dose can be administered safely into the animals. So there are different types of toxicity studies, like acute toxicity study, subacute, chronic toxicity studies. Acute toxicity study means it is with a single dose administration. It's called as single dose studies. That is called as a acute toxicity study. The repeated dose toxicity studies are either subacute or chronic toxicity studies, depending upon the duration we administer. And there are other types of toxicity studies like genotoxic studies, carcinogenicity, oncogenicity, hypersensitivity studies, and toxicity on the local surfaces, local toxicity studies. Coming to acute toxicity study, it is the toxicity which results with a single dose of drug administration. So for conducting acute toxicity study, we usually take two species of animals. Out of these, one must be a rodent, another must be a non-rodent. So observed, the toxic symptoms are usually observed for 3 to 14 days, depending upon the type of drug and for what it is meant for. And in this case, acute toxicity study, we record the mortality and other changes like morphological changes, biochemical changes, pathological, histological. The dead animals are to be investigated, and we can determine the LD50 dose from these acute toxicity studies. That is the major importance of acute toxicity studies. So within 24 hours, the animals means the dose which kills the 50% of the animals in the group, that is called as LD50 dose. And from graphical method and arithmetic method, we can know the dose, LD50 dose. Then there are different guidelines are available for conducting acute and chronic toxicity studies. So usually many of us, we follow the OECD guidelines. OECD is Organization for Economic and Cooperation and Development. So these guidelines are normally followed for all these toxicity studies. So some of the toxicity guidelines I want to give. The test guidelines of 420, which indicates fixed dose or limit test, means if it is a herbal drugs, normally we go for 2000 mg per kg body weight. If the drug is found to be safe at 2000 mg per kg body weight, we consider it is safe. So like that, there is another acute toxicity class method which is described in test guideline 423, and 425 describes up and down regulation method, and test guideline 402, it indicates acute dermal toxicity. 403, acute inhalation toxicity. These are all for acute toxicity studies. For repeated dose toxicity studies, test guideline 407, which indicates 28 days oral toxicity study in rodents, and 408, 90 days oral toxicity study in rodents. 410, 21 or 28 days dermal toxicity study. 411, 90 days of chronic dermal toxicity study. 412, 14 or 18, 28 days repeated dose inhalation toxicity study. 413, 90 days of chronic inhalation toxicity studies. Like this, there are guidelines for OECD for conducting acute and chronic toxicity studies. So chronic toxicity studies, normally two species of animals are used, out of which one must be a rodent, one must be a non-rodent. Test compound is administered for more than 90 days in case of chronic toxicity studies. Then animals are observed periodically, not regularly, not daily. Provides inference about the long-term effects of the test substance. These results may be extrapolated to human safety. The report on chronic oral toxicity is essential for new drug entities. So these are all the toxicity studies. If they are the safety studies, they have to be conducted for knowing the safe dose that is to be administered in animals to know the potency, efficacy of the drug. So normally 1/10 to 1/50th dose of LD50 is considered for administration for testing for efficacy, pharmacological activities. So some of the in vitro antioxidant studies for understanding of the budding researchers. I'm giving here some of the in vitro antioxidant studies like superoxide radical scavenging activity, hydroxyl radical scavenging activity, hydrogen peroxide radical scavenging activity, DPPH radical scavenging activity, nitric oxide radical scavenging activity, determination of reducing power, antioxidant capacity, determination by phosphomolybdenum method. Then acute to chelating activity, ferric reducing ability for power, we call it as a FRAP assay, and the antioxidant capacity determined by radical cation ABTS assay, in vitro glycation of bovine serum albumin, and determination of cardinal content. So from this, we are conducting many in vitro antioxidant studies. I would like to give some of our results. Actually, I don't want to throw our entire research onto you. Just I want to give you some example for better understanding how we did and what the results we got from our studies. This is the antioxidant study of hydroxyl radical, nitric oxide, reducing power. So these are all the studies we have conducted for aqueous pericarp extract of Punica granatum. Punica granatum, one of our PhD students, she worked on this, and her results I'm giving here, and the APPC shown good activity compared with ascorbic acid. These are all the IC50 values I have given here for peroxide, hydroxyl, hydrogen peroxide, DPPH, and nitric oxide, reducing power, and the phosphomolybdenum data. Then some of the other assays like FRAP assay, peak assay, and the protein terminal content. Here also the EPP is shown to have a good activity and a comparable activity with that of ascorbic acid in all these in vitro antioxidant activities. So this is one in vitro study. This is using fog rectus abdominis muscle. We have tested the acetylcholine and the nicotinic effects and the complexes with metal ions we have tested, and that is on normal skeletal muscle as well as in the visceral treated skeletal muscle. That were some of the results of these in vitro studies. Just here I'm mentioning the in vitro studies, how we can do the in vitro studies before going for in vivo studies. So other in vitro studies which include cell line studies. So one of our compound, two of our compounds like berberine, berberine, monoamine methylphenate, MAG. These two were compared with the doxorubicin on HEP2 cell lines and the pancreatic 1C cell lines. So these cell line studies, they indicated that berberine and MAG were found to have good activity against HEP2 and pancreatic cancer cell lines. HEP2 and pancreatic cancer cell lines, these two are. And with these two drugs, they isolated pure compounds, so berberine and MAG, and they were shown to have very good activity against hepatic as well as pancreatic cell lines. So this is some of the results about our in vitro studies. Then coming to in vivo studies, there are different steps involved in vivo studies. The first step is determination of toxicity for the compounds and LD50 determination using acute, subacute, and chronic toxicity studies. Then in the second step, evaluation of pharmacological activities, animal models, development of animal models or use of available animal models for inducing disease or injury. And in the third step, the compounds have to be tested for their pharmacokinetics, like that is ADME studies, like absorption studies. Means after developing a drug, after knowing its efficacy and safety, so we can administer. But before administration of the drug, we need to know its pharmacokinetics also, because after its administration into the systemic circulation, how the drug is behaving, what is happening to the drug, we need to know. So for that, ADME studies like absorption studies, concentration of the drug in the tissues, organ, fluids, means whether there is an accumulation of the drug or a drug is available in the free drug is available in the blood circulation or not, and what ratio it is there, that can be known from ADME studies. And histopathological studies means not only the targeted effect, we also need to know its effects on other organs. For that, we need to have histopathological studies. And the serum estimation of biological indicators for drug or metabolite, so that is, we call them as biomarkers. So biomarkers can also be studied. So This is about in vivo studies. We have conducted some in vivo studies. I would like to present the results of in vivo studies. Here, what we did is we are doing a number of pharmacological activities. We have screened many number of herbal extracts or isolates, herbal isolates for their pharmacological activities. But our main concentration is on the diabetes and other like hepatoprotective activity, anti-lithiatic activity. So for your better understanding, I would like to give some of the approaches, how we have gone and how we have tested our compounds for anti-diabetic activity and what are the parameters we have estimated. From that we can have some idea how to go for a research. Before going into that, why we have concentrated on diabetes is diabetes is a very highly spreading disorder, we can say. So there are two types of diabetes, type 1 and type 2. So we know type 2 is more prevalent than type 1. Type 2 is almost 90 to 95% total diabetes is type 2 only. So according to worldwide diabetes, according to this is Indian Diabetic Federation studies. In 2015, there were in the worldwide 415 millions, and it may be rising to 642 million by 2040. And India also, it is become a diabetic hub now. 2015 only 70 millions, and it is these figures are not correct actually. So it is rising like anything, We know that different types of diabetes, type 1, type 2, and gestational diabetes. There is, you can say, pre-diabetes, that is due to impaired glucose homeostasis. And there are some other specific types, because they are not much prevalent, we may not discuss on that. But our major concentration is on the type 2 diabetes. So this is the type 1 diabetes. So we know that type 1 diabetes is mainly because of the destruction of pancreatic beta cells. So completely there is no insulin, there are no pancreatic beta cells, or there is no insulin to produce to release into the bloodstream to control the high blood glucose levels. That's what the condition of the type 1 diabetes. It is a mainly autoimmune disorder, and the prevalence is 5 to 10% among all diabetes. Type 2 diabetes is, as we know, type 2 diabetes is mainly, there may be insulin, there may be pancreatic insulin present synthesized in the pancreatic beta cells, which may not be released into the systemic circulation. That is one kind of type 2 diabetes. Another kind is, even it is released into the systemic circulation, it may not be effective in controlling blood glucose levels. That is mainly because of insulin resistance. So these two are the major factors which are responsible for type 2 diabetes. So gestational diabetes, as we know, is glucose intolerance with the onset and first recognition during pregnancy. It occurs in the 7% of all pregnancies according to statistics. So does not include women who have diabetes diagnosed before pregnancy. So they cannot be included in this gestational diabetes. They are considered as a diabetic women only. But only gestational means once the pregnancy, means during the second and third trimester, if they are diagnosed for any insulin resistance or diabetes, such individuals are categorized under gestational diabetes mellitus. So there are mechanisms which leads to this insulin resistance. So normally, when there is increased glucose levels, that causes release of insulin secretion. Apart from blood glucose regulation, insulin is also having other effects on other organs, like on the brain. It increases food intake, means more hungriness, and that is may lead to obesity. That obesity may further lead to insulin resistance. Similarly, in the liver, it increases glucose production and the increase in plasma glucose. And even all these are effects of decreased effect of insulin. The insulin secretion is decreased, that impact on these organs. So decreased insulin secretion on the skeletal muscle, it decreases efficiency of glucose uptake, and there is increase in the plasma glucose. On the adipocytes, increases lipolysis, and that causes insulin resistance and decrease in beta cell function. These are all the effects. And there are risk factors for diabetes as well as for diabetic complications. Overweight and obesity is the risk factor for diabetes, and genetic and family history is also one reason for diabetes. High blood pressure and use of drugs for high blood pressure is also one cause for diabetes and gestational diabetes. So these are the factors which are responsible for diabetes. And the diabetes itself, it causes the complications when there is increased blood glucose levels in the body. So that increased blood glucose levels are maybe converted into some other toxic forms and leads to diabetic complications like retinopathy, neuropathy, nephropathy, cardiovascular diseases, and amputations. These are the basics. I'm not don't want to go in detail about all these because many of you know. So the molecular mechanism how the diabetes leads to is one mechanism is generation of reactive oxygen species, ROS. Reactive oxygen species are generated because of obesity or because of high stress. These are reactive oxygen species are generated. Even for those who are overeating, that also causes overstress, and that also causes reactive oxygen species generation. And another mechanism is formation of advanced glycation end products, AGEs, in the body. AGEs are formed normally when the proteins, the amino acids, they react with the excess glucose that forms AGEs, advanced glycation end products. Then altered incretin gene expression. I will discuss more about this incretin gene expression. What are incretins in later my slides. Then activation of polyol pathway. This is one pathway which converts the excess glucose into sorbitol and fructose, and those fructose may deposit on the muscles or neurons, and they may cause myopathy, neuropathy, like that. And altered glucose homeostasis. This is another mechanism which may lead to diabetes or diabetic complications. Induction of flux through exosomal pathway. This is another pathway which is responsible for diabetic complications and obesity. So these are all the mechanisms which are mechanisms of diabetes which may cause increase in the diabetes or diabetic complications. So normally in the animal models we do anti-diabetic activity. Majorly we take two models. One is STZ-induced diabetes, another is alloxan-induced diabetes. Many of you might not be knowing how these drugs they produce and what is the difference between STZ or alloxan in inducing diabetes mellitus. So STZ mainly it induces diabetes by destruction of beta cells in the pancreas. So directly STZ that it destroys the beta cells and that causes necrosis. That the necrotic beta cells are engulfed by the macrophages, and they are the beta cell mass will be reduced. When the beta cell mass is reduced, that causes insulin insufficiency. The sufficient insulin cannot be released from the pancreatic beta cells, so that leads to chronic hyperglycemia. This is otherwise referred as a residual beta cell population. So this is how directly the STZ, which directly destroys the beta cell function, and thereby it reduces the insulin insulin secretion, and it leads to chronic hyperglycemia. Whereas alloxan it cannot act directly on the pancreatic beta cells. It causes generation of free radicals. So the alloxan is reduced by reduced glutathione. The glutathione gets oxidized, and the alloxan gets reduced to dialluric acid. This dialluric acid, by auto-oxidation, it is converted into alloxan radical. This alloxan radical, in presence of oxygen, it forms reactive oxygen species. That is O2 molecule, so that reactive oxygen species. These reactive oxygen species can be neutralized by SOD, superoxide dismutase, which is endogenously secreted antioxidant enzyme, so which neutralizes these oxygen free radicals into H2O2. So H2O2 is unstable. So this H2O2 by Penton reaction, it releases a number of hydroxyl radicals. these hydroxyl radicals, by acting on the beta cells, they destroy the beta cells. So this is how the alloxan induced diabetes is mostly by free radical generation, whereas STZ induced diabetes is by direct damaging of the beta cells by reducing the beta cell mass. So that's what the difference between these two mechanisms. So mostly people follow these two models for induction of diabetes in animal models. So this is about the production and metabolism of reactive oxygen species. We know that there are many reactive oxygen species that are generated in our body. Mainly the oxygen free radicals are first generated. These oxygen free radicals, by combining with nitric oxide, they form peroxynitrile, and then some of these oxygen free radicals are neutralized by superoxide dismutase. This neutralization is that forms unstable H2O2 molecules. These unstable H2O2 molecules further, by Canton reaction, they are converted into hydroxyl free radicals, which mainly in case of alloxan, these hydroxyl free radicals only they cause the damage to the pancreas. And another two endogenously released enzymes are catalase. Catalase, some of it converts the unstable H2O2 into H2O and water, and thereby it reduces the amount of H2O2 in the body. And similarly, glutathione peroxidase, which also converts H2O2 into H2O and oxidized glutathione. So this myeloperoxidase uses the chloride ions. Chloride ions reacts with H2O2 and that forms H2O. That's how this SOD, catalase, myeloperoxidase enzyme, and glutathione peroxidase, all these enzymes, so they are the endogenously released antioxidant enzymes. They protect us from the oxidative damage. So there are enzymes like enzymatic reactions like cyclooxygenase, lipoxygenase, uncoupled MOS, all these xanthine oxidase. They mediate all these. They involve in the generation of free radicals, and also by non-enzymatic reactions like mitochondrial respiratory chain, glucose autooxidation, polyalpha. They release the oxygen free radicals in the body, and they can be scavenged or they can be neutralized by the endogenously produced antioxidants. So these are some of the results of our studies on endogenously released antioxidant enzymes. So these are this is a control, diabetic control, and we have taken glycoside as a standard drug. This is a pericardium with a extract of Peronia limonia, 50 mg and 100 mg. So you can see the statistically significant protection, means increase in the levels of SOD, catalase, BPX, GSH, and the EST levels from our studies. This is one example how we did conduct the study, how we justified our study. And coming to the advanced glycation and products, so this is another kind of study. During diabetes, what happens? The diabetes generates advanced glycation and products. These, when there is a increased glucose in the body, in the system circulation, it reacts with the amino acids. So by reacting with amino acids, it forms shift base. This shift base is formed within hours to days. These shift bases are converted into amidori products. Amidori products by within days to weeks, and here only the hemoglobin HbA1c are formed in the amidori products. These amidori products are further converted into advanced glycation products. So these advanced glycation products are formed within weeks to months. So these advanced glycation products, some of these are CML, pyrroline, metocidine. These are the advanced glycation products that are formed from the glucose and amino acid reactions. This is how the AGEs are formed in the body. Similarly, there are other pathways. This glucose can be converted into sorbitol and fructose by polyol pathway. So first of all, it is converted into sorbitol, then fructose. This fructose 3-phosphate and its other metabolites, that increases alpha oxaldehydes. These are methyl glyoxal, 3-deoxyglucosamine. These are also called as advanced glycogen. They increase the formation of advanced glycogen products. Similarly, lipid peroxidation in the body also helps in the formation of all these products. And glucose auto-oxidation, directly glucose auto-oxidize, and that also produces these methyl glyoxal, 3-deoxyglucosamine. All these are collectively responsible for formation of advanced glycation end products. These advanced glycation end products are sometimes they are reversible, but most of the times they are irreversible. So these advanced glycation end products, when they are deposited on the tissues, or they may be deposited on the neurons, they may be deposited on smaller blood vessels like capillaries, and they form like neuropathy, nephropathy. So like this, all these diabetic complications are mainly because of the formation of advanced glycation end products. This is how the diabetes leads to diabetic complications. Here, some of the results we have studied for AGE levels using the extract of lychee chinensis on the serum, lens, and renal AGEs. So the serum AGEs, lens, eye lens, and kidneys. The lens homogenate and kidney homogenate also we have estimated the AGE levels, and these are the results that are we have incorporated here. Similarly, the PCO levels, that is in the serum, lens, and kidney. So these are the our study results of estimation of AGEs in serum and other organs which are affected by induction of diabetes. So the diabetes is maintained for I think more than 8 weeks, I think, and that is responsible for the development of diabetic complications. So coming to the polyal pathway, as I discussed earlier, the polyal pathway increases glucose will be converted into sorbitol by aldose reductase. This sorbitol by sorbitol dehydrogenase, it is converted into fructose. So this fructose is actually it is not reversible. This fructose may induce the generation of advanced glycogen products. So this is how this polyal pathway is one kind of pathway which is responsible for diabetic complications. So on polyal pathway also we have tested punica granatum extract. Punica granatum at 2 different dose levels it has shown to produce good activity compared to this by estimating the aldose reductase enzyme and also sorbitol content we have studied. These two are the parameters which indicate the polyal pathway. So that's how you can take the different parameters to estimate different pathways. By that parameters, you can see how the diabetes or diabetic complications are occurring and how the extracts or compounds are preventing these diabetic complications. Here, another interesting and important study what we have conducted in our laboratory. I would like to impress you upon this study. This is regarding the incretin effect. Some of you might be knowing about incretins. So incretins are the gastrointestinal enzymes. There are 2 incretin enzymes. They are called as 2 incretins, GLP-1 and GIP. GLP-1 and GIP. Here you can see in the intestine, small intestine. So in this L cells of what we can say before colon, ileal part of the intestine, there are L cells. In the L cells, proglucagon, which is a precursor for GLP-1 and GIP, proglucagon is present. This proglucagon, which is converted into glucagon and mini glucagon by prohormone convertase 2. And the same proglucagon is also converted into GLP-1 by prohormone convertase 1/3. So this is how the GLP-1 is formed. So another incretin hormone is GIP, which is formed from pro-GIP. So from pro-glucagon, pro-GIP is formed, from pro-GIP, GIP is formed. These GLP-1 and GIP, they by acting on the receptors on the pancreatic beta cell. So remember, the insulin which is released from the pancreatic beta cells with respect to high blood glucose levels, okay, is not directly releasing. There is influence of GLP-1 and GIP on these receptors that are actually motivating the release of insulin from the pancreatic beta cells. So these are also glucose dependent only, GLP-1 and GIP. So GLP-1 is more potent compared to GIP in synthesizing, secretion of insulin. So it also helps in the synthesis and the secretion of insulin. Remember, there is a pro-glucagon present in the pancreatic beta cells also. That pro-glucagon produces glucagon in the pancreatic beta cells. Similar pro-glucagon which is present in the alpha cells, which produces GLP-1 and GIP. So I'll tell you about the GLP-1 and GIP, which are considered called as the incretin hormones. The role of active GLP-1 and GIP in glucose homeostasis. So what they do is, GLP-1, GIP are secreted in response to food intake. So whatever the food we take into the gastrointestinal tract, depending upon the quality and quantity, the GLP-1 and GIP are released to the gastrointestinal tract. So they cause stimulation of insulin biosynthesis and secretion. And GLP-1 inhibits the glucagon secretion from the pancreatic beta cells, pancreatic alpha cells. And the GLP-1 and GIP, they decrease the blood glucose levels. That's how the GLP and GIP are helping in reducing blood glucose levels. This is completely GIT mediated incretin hormone activity. So this GLP-1 and GIP, they are unstable. Their activity is only for about 3 to 4 minutes, particularly the GLP-1, which is very unstable, which is degraded by dipeptidyl peptidase 4 enzyme. So this dipeptidyl peptidase 4 enzyme, it immediately metabolizes GLP-1. So we know the drugs like recent anti-diabetic drugs, sitagliptin, all these gliptins, vildagliptin, sitagliptin, all these gliptins, they act by inhibiting the DPP-4 enzyme, and they increase the GLP-1 levels, and further they increase the release of insulin, and that lowers the blood glucose levels. That's what the mechanism of DPP-4 inhibitors. And there are some GLP-1 analogs are also were there in the market, like exenatide and other compounds are similar to exenatide, and which they are called as GLP-1 analogs, which are also there. But the GLP-1 analogs and the DPP-4 inhibitors are having major adverse effects. And because of their major adverse effects, their usage is limited. Then these are some of the what we did is in our study. Our intention was most of these plant-based drugs, because all these plant-based drugs we consume, we take by oral route only. So they are meant for oral route only. So when they are administered by oral route, so we have tested whether they are directly acting on the pancreatic beta cells or they are acting through some other mechanisms. So that our interest is to know their effect on these incretin hormones in the gastrointestinal tract. So we have tested for their activity on the pro-glucagon levels, DPP-4 levels, GLP-1 levels, and even insulin receptor levels by gene expression studies. So some of the results I am presenting here of aqueous extract of Garcinia cambogia. This is you can see here, this is insulin receptor levels were found to be increased with the treatment with the Garcinia cambogia. This is with the this is about proglucagon levels. So the proglucagon levels were also found to be increased compared to the diabetic control with the plant extract treatment. And that means the precursor for proglucagon. So another interesting finding, our finding is when we induce diabetes using STZ, there was complete absence of proglucagon levels what we found in one of our studies. So that means whatever the induction that is causing, even in diabetics also, there might be a reduction of proglucagon levels in the gastrointestinal tract, which might be responsible for decreased incretin hormone secretion, and which might be responsible for decreased insulin secretion, and which was found to be recovered by using these plant extracts because they are meant for oral administration. So this is about a DLP expression. So you can see here how the DLP levels were increased the treatment, and this is a DPP4 expression. DPP4 expression was found to be reduced with these drugs. Means they are not only improving the precursor for GLP-1, they are also inhibiting the DPP. Some of the drugs are also inhibiting the DPP4 enzyme. They are actually similar to the of like DPP4 inhibitors, sitagliptin, vildagliptin, all those drugs. Then coming to the glucose homeostasis, we know you can understand increase in the blood glucose levels that causes release of insulin from the pancreatic beta cells, that increases the glucose uptake by the adipose tissue as well as the skeletal muscle, and that increases glucogenesis, gluconeogenesis, and the uptake and utilization that decreases blood glucose levels to normal level. Okay, so normal glucose levels are maintained between 4 to 6 millimoles. When glucose levels are reduced, that causes stimulation of pancreatic beta cells for the release of insulin, and there is a release of glucagon also, which causes glucogenolysis and gluconeogenesis that causes blood glucose, increase in blood glucose from normal. When increase in blood glucose is there again, it causes release of insulin. That's how the glucose homeostasis is maintained in the body. So these pathways are also tested, like we have estimated the glucokinase enzyme, lactate dehydrogenase enzyme, thyroid kinase enzyme. The glucokinase enzyme is measured with respect to G6 phosphate liberated per minute per mg of protein. Similarly, lactate dehydrogenase is measured by Parmesan formation per minute per mg of protein. Similarly, by pyruvate kinase by measuring the pyruvate formed per minute per mg of protein. That's how we have estimated the enzyme levels also, and glucose 6-phosphate levels, fructose 1,6 biphosphate levels. So like this, many parameters we have studied in our anti-diabetic and as well as compounds for diabetic complications. So this is about obesity and diabetes, the interrelation between obesity and diabetes. Obesity normally causes insulin resistance. So obesity increase in beta cell function, increase in beta cell growth, normal glucose tolerance, compensatory hyperinsulinemia. So beta cell dysfunction leads to beta cell apoptosis, impaired glucose tolerance, and that leads to type 2 diabetes. That's how the obesity leads to type 2 diabetes, because of the impaired glucose tolerance that causes hyperinsulinemia as well as hyperglycemia. You can observe in case of type 2 diabetes, there will be increased insulin levels in the blood and increased glucose levels. That indicates insulin resistance. That's how we can know whether it is type 1 or type 2. And other enzymes, other like parameters which also supports these anti-diabetic studies are estimation of triglyceride levels, total cholesterol levels, the HDL levels, LDL levels, VLDL levels. Usually HDL and VLDL levels are calculated, and these not only these, we have also estimated directly the insulin levels and insulin resistance, HOMA-IR, insulin resistance levels also we have determined by using different methods. This is how the complete study about the diabetes and the diabetic complications. The different extracts were evaluated for their anti-diabetic activity and their beneficial role in the prevention of diabetic complications. Some histopathological reports of these anti-diabetic studies. So if you see the pancreas, you can see the pancreatic beta cells here. This is the control animal pancreas. This is the disease control, how the destruction is produced in the diabetic control. Similarly, these are the standard how it is improved the beta cell function. This is with one of the compound like Rhamnus persiana extract we have tested. These are some of the results of histopathology of pancreas. Similarly, histopathology of testis, you can see the testis histopathology process. So this is with the control. This is one control. You can observe the damage to all these basement membrane, and you can see this is with the standard. Again, there is an improvement. This is with another extract, cassia orculata 200 mg and cassia orculata 400 mg. You can see all these parameters in the histopathology of testis. Similarly, histopathology of ileum, we have also tested the ileum histopathology because what we felt is because the drugs are acting directly on the ileum part and the L cells of the ileum, and that is responsible for the secretion of GLP-1 and GIP. So we thought of going for a histopathology of ileum, and we found that there is a damage to ileum in the disease control, and this was found to be recorded with the standard drug as well as with the extracts that were administered by oral route. You can see all these microvilli, all these here. Then histopathology of retina of the eye. You can see this is the control. Here, this is control standard and treatment with the Memodica pupincinensis. This is another extract we have tested. So you can see number of plants we have evaluated for their anti-diabetic and against diabetic complications. This is the slit lamp technique, and probably we have done. I don't know whether other people are doing or not. So this is by taking the help of one of the ophthalmologist who helped us in taking these pictures using slit lamp of the rat. It is the control. You can see that it is clear eye, and this is the control. This is control. This you can see the opacity here. This is the standard treatment. This is with the tunica granatum, and this is with the coronial pneumonia. So here we have given the different stages. So clear means it's very clear. Then stage one means vacuoles. Here you can see this is the phase 3 to 4. This identification was given by the doctor only. Vacuoles, and this is a stage 2 means a cortex haziness. So here it is 0 to 1, that means clear to vacuoles. Opacity is present, is 3. You can see the opacity here. It's a mature, means completely opaque, no visibility. That is a mature cataract. So stage 4 means complete mature cataract, which was observed with the chronic induction with the diabetes. We can observe the mature cataract also. We have also studied the fundoscopy of the eye. So this is the control. You can see the disease control, complete absence of fundus. This is again with the standard treatment. This is pericarp extract of tunica granatum. This is with the coronial pneumonia. So this is like different studies we have conducted. This is with the seminal morphology study. Hello. We have estimated the semen means morphology, means semen count, sperm count, and sperm even motility also. Sperm characteristics we have studied with in case of control and diabetic control and treated. You can see some of the sperms here. So this is about the diabetic, means almost complete all organs we touched, and we have taken all parameters in case of diabetes, and we have tested for their activities. Similarly, we have evaluated many compounds for hepatoprotective activity. So normally, for better understand how the hepatoprotective activities, after hepatic damage is induced, normally we use paracetamol, ethanol, CCl4, and isoniazid, and other anti-tubercular drugs. Some of the anti-tubercular drugs can be used as models for induction of hepatotoxicity, in particular in rat model. So ethanol consumption that by we know ethanol is converted into acetaldehyde and then it is converted to acetate, and which causes damage. Normally the steatotic liver is caused because of ethanol consumption. So this in vitro testing, there is increased reactive oxygen species, increased nitric oxide, increased enough NRF2. So like this, all these can be measured. Antioxidant agents can be measured in vitro. We can test, and in vivo also you can measure the enzyme levels. And in case of acute and chronic liver toxicity, the parameters that can be studied: increased creatinine, total cholesterol, increased fraction, HDL fraction, reduced oxidase ratio like SOD, and reduced glutathione peroxidase, increased T bars, protein carboxylated carboxylation increases, MMP2 levels are increased. In case of acute chronic means urea, creatinine, increase in ALT, decrease in HDL, increase in THC, decrease in catalase. Like this, these parameters can be considered for testing the hepatoprotective activity. These are the models and the parameters. And other parameters you can see in case of hepatoprotective activity, you can take the parameters like SGOT, SGPT, alkaline phosphatase, albumin, bilirubin, alpha glutamyl transferases, lactate dehydrogenase. Why I'm giving all this data is to understand that whenever you go for a hepatoprotective activity, what kind of parameters can be estimated and where they can be estimated. Is to know, to have better idea about all these parameters. I'm giving here some of the parameters. Similarly, antioxidant parameters like total protein, SOD, catalase, DSH, GR, EST, GPX, MDA, all these levels can be estimated in the tissue levels. Even in serum also you can estimate, but the tissue protein, all these can be estimated. And apart from this, hepatic injury means there is involvement of inflammation. Definitely there is a generation of inflammatory parameters like DNF alpha, interleukins, CRP, and also you can conduct the histopathological studies. This is how the parameters, the different parameters can be estimated at different levels, and you can check whether your compound is how much it is effective against all these parameters, what parameters it is increasing, how it is protecting the liver. That can be known from these parameters. And some of the results I am presenting here of hepatoprotective activity, you can see here we have studied with the different plant extracts and polyherbal mixtures we have prepared. And the polyherbal mixture also of these, they have shown very good activity against bilirubin, albumin, GGT, LDH. These are all the parameters what we have estimated. Similarly, here also using other extracts, polyherbal mixture. So these are some of the results: DSH, DPX, DSP, and GR. And other activity is anti-lithiatic activity. So against ethylene glycol induced lithiasis. As we know, lithiasis is induced. The one efficient model is ethylene glycol. So the ethylene glycol, how it induces is ethylene glycol. It can be converted into glycoldehyde by alcohol dehydrogenase, and glycoldehyde by aldehyde dehydrogenase. It is converted into glycolic acid, and the glycolic acid is converted to glyoxalic acid, and this converted to oxalic acid, and oxalic acid by combining with the calcium that forms calcium oxalate crystals. These crystals are deposited. So this is called hypocalcemia. Renal tubular necrosis is caused by these calcium oxalate crystals. This is how the mechanism, how the ethylene glycol produces lithiasis. So lithiasis may be anywhere. Maybe lithiasis in the kidneys, and maybe the pancreas, or maybe the ducts. Like that, lithiasis can be formed anywhere in any organs, and these calcium oxalate crystals may be formed. The mechanism will be the same. So the parameters here, what we can estimate to know the lithiatic activity. So crystal parameters in the blood or serum, calcium levels, oxalate levels, uric acid levels, crystal inhibitor levels like magnesium and citrate, Other parameters like bilirubin, creatinine, and urea, depending upon the organ where they are deposited. So you can choose the parameter. The urine parameters like calcium oxalate and uric acid in the urine, and the urine volume, pH, and glomerular filtration rate, microscopical examination of the urine, and tissue parameters like oxidative stress parameters like SOD, catalase, GSH, PCO. Even here also the inflammation is produced, the inflammatory mediators are released. The inflammatory parameters like interleukin 6, 1, IL-12, 23, and histopathology of the organs where the crystal deposits are there like pancreas, kidney, and bladder. So this is how the lithiatic activity can be taken up. So these are some of the results of anti-lithiatic activity in lithiasis induced using glycol in rats. So this is the creatinine levels, uric acid levels, urea, calcium, and oxalate. This is the kidney parameters. All these kidney parameters is lithiasis, urolithiasis, and here the microscopy analysis of urine. Here the crystals are not clear, but we got very good pictures. If you see under the microscope, you can observe very good crystals. This is in case of a disease control. We found good crystals in the urine and how they are formed. And standard, and there is no appearance of crystals, and the treatment also, the crystal number was found to be reduced. And if you see the histopathology of kidney, we can conduct the histopathology also. This is the Bowman's capsule, and you can see how the spaces are affected in case of disease control. This is control, and this is disease control. You can compare between control and disease control. And this is histopathology of the bladder, urinary bladder. So because of the crystal form, they may cause the damage to the urinary bladder, and the urinary bladder histopathology also we have taken into consideration. And then next activity is dextran sulfate sodium induced, DSS induced inflammatory bowel disorder, IBD. So IBD also we have studied some of the extracts, some of the compounds we have studied for their activity against IBD. So IBD was induced using the DSS model, 35 to 50 kilodalton DSS, about 1 to 4% was mixed in drinking water, administered orally to mice for 7 days. That is the standard method, and which produced damage to the colonic epithelium. And we found this is the mechanism how it produces colitis, impaired cytoskeletal structure of colonic epithelium, infiltration of into the inflammatory cells, and the induction of colitis. And it is also by mitochondrial dysfunction, increasing oxidative stress, cell cycle arrest, apoptosis, and that also leads to induction of colitis. These are the parameters what they've increased in case of DSS induced inflammatory disease, IBD indicators. These are the TNF alpha, interleukin 1 beta, interleukin 6, EPC, myeloperoxidase levels will increase, and the colon length will decrease, SD levels will decrease, and the catalase. So SOD catalase levels are also decreased. These are the parameters that can be estimated in case of DSS induced IBD. General condition, we can know the length and the weight of the colon, weight of animals, fecal occult blood, nature of feces. So this is can give the score also. And in the blood parameters like CBC in the blood, CRP, C-reactive protein in the blood serum can be estimated. And the tissue parameters like oxidative stress parameters like catalase, GSH, PCO, MDA, nitric oxide, and SOD. The inflammatory parameters like interleukin 6, 1 beta, interleukin 12, 23, all these they indicate the dominant means IBD. They are all expressed in case of IBD. And advanced glycation and products like CML. So remember, even in case of IBD, CML is expressed. Okay. So CML levels can be estimated. These are all the parameters which indicate the induction of IBD by DSS. So these are some of the results. The rectal bleeding you can observe. This is the intestine, how that shrinkage of the intestine. You can check the length of the intestine also in case of ulcerative colitis. So length and weight of the intestine. is an observed here the same case with the DSS induced coitus group, and this is with the berberine treatment. Last one, berberine treatment was found to again normalize, protect. Sorry, this is a protective activity found to protect the DSS induced ulcerative colitis. So these are the some of the parameters: interleukin 6, 1 beta, 12, 23, CML levels were estimated. Disease control, and 50 mg and 100 mg of berberine. So the histopathology of colon under normal conditions. If you see the DSS induced colon, you can observe the histopathological changes in the colon. Then another is cardiovascular system. We know that cardiovascular system, this cardiac can be known. Cardiac function can be estimated by ECG, and can be means there are screening methods that can be used: a manual patch clamp technique, automated high throughput patch clamp, isolated organ filtration, whole heart filtration, isolated Purkinje fibers. There are so many kind of models are available to know the cardiovascular means to screen any drug for cardiovascular activities. For hypertension also there are models available. For heart cardiac activity also, cardiac muscle activity also, there are different models that are available. So these are the different means. What I want to say to you is this is all. These are the different experimental models, and how we can test our compounds for their activity against different models, and what parameters we can estimate. So with our results are given. And another interesting study, the most of our means lab is many of our students, they are engaged in drug-drug interaction studies. I think very few people are conducting drug-drug interaction studies. Our lab, I think it's known for drug interaction studies. So how do we plan the drug interaction studies? Plan of the study is conducted in two dissimilar species. So this is like a toxicity studies only. So we take one rodent and one non-rodent model. For rodent we take rat, and for non-rodent rabbit. So rat, why we take rat is a small animal, easy to handle, and the metabolic pattern and blood glucose levels, blood pressure levels are more or less similar to humans. They can be induced to diabetes very easily, because we have tested the anti-diabetic drugs interactions with other drugs majorly. And the rabbit is a comparatively high animal, more number of blood samples can be taken, and it is the model for bias of insulin. That's why we have chosen rabbit for drug interactions of anti-diabetic drugs. So our interest is if the interaction occurs in two dissimilar species, it is likely to be the same in humans also. That's with that objective, we conduct drug interaction studies. So pharmacodynamic studies usually conducted in normal and diabetic rat model, pharmacokinetic studies in normal rabbit model. So some of our drug interaction studies, so we have conducted drug interactions between many drugs which are used normally in case of diabetes, means diabetic drugs with other co-administered drugs. Normally, as we know, diabetes is a complex disorder. Diabetes is also associated with other disorders like cardiovascular and many other disorders like retinopathy, nephropathy, neuropathy. So once diabetes is there, means it may lead to chronic diabetes, may lead to many other complications. In such cases, anti-diabetic drugs are to be combined with other drugs. So in such cases, there may be possibility for drug-drug interactions. So such drug-drug interactions work. We have established for their safety, like we have taken clozapine and risperidone here. These are all these two are the antidepressant drugs. We have tested for their safety. This is along with glycoside, and here we have also tested some of the plant products also with anti-diabetic drugs for their safety, and we have established the safety of these some of the plant extracts, and even some of the formulations we established their safety in presence of the anti-diabetic drugs. So we have also studied the pharmacokinetics here, how the means whether that interaction is of pharmacodynamic nature or pharmacokinetic nature. Do not know that mechanism of interaction. We have conducted the pharmacokinetics also. So these are means I don't want to go in detail about all these again. This is about the clinical trials. Some of our students are engaged in clinical studies from the fifth year and the sixth year students, and also our pharmacy practice students. They go direct regularly to the hospital where they do some studies. Most of them are base studies only. So these are some of the designs of these clinical studies, like analytical descriptive studies, analytical again experimental and non-experimental studies. Experimental studies again randomized clinical trials and non-randomized clinical trials. We never involved in till now. We have not involved in any experimental studies. And non-experimental studies, we have conducted some cohort studies, case cohort studies, case control studies, cross-sectional studies, and descriptive studies. Many of our students, they are doing descriptive studies like surveys. So this is one of the descriptive study. The percentage of incidence of this, I think it is breast cancer in rural and urban population. We have evaluated in Visakhapatnam district. Around 100 people we have taken, and urban people were less compared to the rural people. So like this, some reasons why they are we could able to find some of the reasons for these cancers and other disorders from these descriptive studies. So this is another study, the patient utilization pattern of the different anti-cancer drugs, chemotherapy, radiation therapy, surgeries, and supportive care. So how the utilization is means people, how many, what is the percentage of people that are on chemotherapy and radiation therapy. So that we have studied one, that is one study. So like this, we are also conducting some clinical studies. Then this is about advances in studies. So now whatever the results I have presented, the results are from our studies, and also we have a collaboration with University of Malaya, Department of Physiology, University of Malaya, Malaysia. So they are taking our samples over there, and they are testing for further studies, and we have published about 10 papers together. We are conducting some basic studies here and advanced studies we are conducting there. So in that they are conducting immunohistochemistry studies and immunofluorescence studies. That's what I have given here, how they are conducting. And so this is about virtual screening and discovery, the advances in the research. Use the combinatorial chemistry, molecular modeling, in vitro, in silico, edema, whatever I have discussed, the traditional research. This is all these are advanced research. That's what it is going on there in University of Malaya. They are all having the facility for all these studies. Then proteomic studies. Some of the proteomic studies can be done. In case of cancer, tumor, vascularization, malaria, inflammation, these are some of the parameters, the targets for all these disorders or diseases. Genomics, molecular modeling, virtual screening. I don't want to discuss all these things. So, actually, to say these are all the advances in the research, I have given all these. So, this is our pharmacology research lab. This is our exclusively for our research only, pharmacology research only. So we got funds from UGC and AICP, and also we got funds from the industry. We are taking up some of the projects of the industry. I'm the advisor for one of the industry in Hyderabad, Suresh Pharma Private Limited. Another also we have associated with other industrial works. And these are our some of our publications. So you can see we have published our drug-drug interactions and other like commercialization of plant extracts for other activities, our girls, and also in collaboration with the University of Malaya and also the industry. So many of our students, they have published in very reputed journals. So all these are journals with a high impact factor only are given here. Thank you. So this is about
It was a wonderful session, sir. Thank you very much for your excellent presentation.
thank you, sir. Thank you, sir.
Sir. So in this session, we have observed the trends in pharmacology research, past, present, and future. Sir has explained the basics and the fundamentals of pharmacology and delivered ideas on different research studies, as well as the content and depth of evidence. Sir, very clearly explained that all your research outcomes with the potential evidences. The session was very interesting and very informative, sir. Thank you very much for your excellent presentation.
Thank you, sir. Thank you very much. Sir, if there are any questions, I'm ready to answer.
Yes, sir. Yes, sir. Yes, sir. I request the participant to come forward for the discussion. Now it's time for the discussion time. Dear participants, the time for discussion.
I think it is a very lengthy. I think it took almost more than 1.5 hours. Maybe they got bored.
No, no issues. No, not like that. Even this was interesting. I was keenly observing all the slides which were presented.
Okay, okay.
Beginning there was a slight interruption was there, then after some time it was very clearly delivered.
Right, sir. Right, sir.
No interruption. Dear participants, any discussion?
Or else you can contact me or mail or my WhatsApp number that is having.
Yes, sir.
So you can send the questions. I can answer at any time, no problem.
Yes, sir. Yes, sir. Sure, sir.
So like that also we can do. I think some disturbance is there. I think they could not able to connect.
Sure, sir.
So,
Madam, Rajshri, madam. Rajshri, madam. Rashmi madam. Oh, I think nobody is showing asking any doubts. No problem, sir. If there any, then we will collect it and send by email, sir.
Right, sir. Right, sir. Okay.
Thank you.
Thank you, Dr. Ishwar Kumar, sir, for it. It was a very informative and interesting session. Thank you for the time and effort you spent for us. Thank you, sir. On behalf of Nehru College of Pharmacy, I thank you, sir.
Thank you very much for. Thank you all of you for patient listening. All the
Thanks
participants.
a lot, sir. Thank
I extend my thanks to the organizers and the principal, Dr. Sudhakar, and the management. Thank you, one and all.
Thank you, sir. Participants, thank you for joining for this session. Next session will start in 5 minutes. I request all the delegates to continue on the meeting. Next session will start within 5 minutes. Nehru College of Pharmacy. Special thanks to all delegates joined for this session. I request all delegates to be continued here for the next session. Once again, a warm good morning to one and all. Thank you for joining for today's session. I request the audience to kindly mute your mic and turn off your cam for a good session. Please do not present your screen also. Kindly cooperate. I request all the delegates to be continue here. We are having a live session from another eminent speaker.
So what are the difference?
Hello.
Professor Dr.
You're
Shishir
audible,
Nandi,
sir.
good morning, sir.
Good morning. Very good morning. Very good morning.
Sir, shall we start, sir?
Yes, yes, I am ready.
Thank you, sir. Professor Dr. Shishir Nandi, former European Union Marie Curie postdoctoral fellow at National Institute of Chemistry, Serbia, Europe. Currently working as a professor and HOD, Department of Pharmaceutical Chemistry in Global Institute of Pharmaceutical Education and Research, Uttarakhand, India. Dr. Shishirnandi completed PhD from Indian Institute of Chemical Biology, Kolkata as a CSIR Gate Fellow and had been awarded PhD in Pharmacy degree 2011 by Jadavpur University, India. He did his postdoctoral research on prediction of new promising cycloaddition reactions, quantum chemical calculations and reactions modeling at transition state levels using different basis sets and theory under the European Union Laboratory of National Institute of Chemistry, Slovenia, Europe. His specialization is pharmaceutical chemistry. Pharmacy is based on the area of pharmacoinformatics related rational drug design, which includes 2SAR and drug design, biological activity prediction of lead compounds, ligand receptor interactions, virtual screening, combinatorial library design, and lead hopping. He published more than 60 original research articles and reviews in reputed international journals having high impact factors. He presented his research at many international conferences around the world. So, is the reviewers of many research articles of medicinal chemistry research Journal of Bioanalysis and Biomedicine, has drug development technologies, combinatorial chemistry, and high throughput screening. Arabian Journal of Chemistry, lectures in drug design and discovery, current medicinal chemistry, etc. He is the guest editor of current signal transcription therapy and current pharmaceutical design and editorial advisory board of Journal of Bioanalysis and Biomedicine. Advanced Techniques in Clinical Microbiology, Journal of Computational Methods in Molecular American Pharmacy, is in microbiology and associate editor of open access medicinal chemistry. International Journal of Pharma Research and Health Sciences, Bioequivalence and Bioavailability International Journal. He is having more than 12 years of research experiences. His research interests in pharmacoinformatics-based QS-AR and drug design, molecular docking, scoring function analysis, computational tools for drug discovery and design, lead optimization, virtual screening, combinatorial library design, lead hopping, quantum chemical calculations, and reaction modeling at transition state levels using different basis states and theory. We are pleased to have you here for the Faculty Development Program conducted by Nehru College of Pharmacy. Welcome you, sir. Sir, audio.
Hello.
Sir. Now it is audible.
Acha, it's audible now.
Yes, sir.
Thank you very much. Thank you very much, ma'am, for such brief introduction. I convey my cordial greetings to the management and director of Nehru College of Pharmacy. I convey my hearty thanks to Professor Sudhahar, program coordinator, who invited me to deliver this talk. You know, everybody is under the threat of COVID-19. It's a big loss. Loss of economy, loss of lives, and loss of national integration of each country. So,
Sir, excuse me.
yes.
Sir, kindly increase the volume in your system. Yeah, please increase the volume in your system.
Hello?
Yeah. Now it is audible, sir. Very clear.
Can you hear me? Hello?
Yes, sir.
Okay.
Yeah. Audible.
So can I continue now? Hello?
Yes. Proceed, sir. Yeah. Continue, sir.
Everybody is under the threat of COVID-19. It's a big loss. Loss of economy, loss of the lives, and loss of national integration of each country. So, how to combat COVID-19? This thing I would like to discuss today. To repurpose of chemotherapeutics to combat COVID-19. It's an original research which I would like to present today. You know what is COVID? COVID is coronavirus diseases. It's not the new one, it's very old diseases. First, it was erupted in 1930 in poultry chicken. For animal, the symptoms are respiratory, gastrointestinal, liver and neurological disorders. Then, severe acute respiratory syndrome caused by SARS-CoV strain occurred in 2003 in China, followed by the outbreak of Middle East respiratory syndrome caused by MERS-CoV strain in the year of 2012 in Saudi Arabia. That time, it was pandemic, and the symptoms in humans are severe acute respiratory syndrome, high fever, pleural effusion, and death. In the end of 2019, again a new strain of coronaviruses erupted in China, Wuhan, China. So it's a new strain which is called as novel coronavirus strain or novel coronavirus, and WHO, World Health Organization, it gives the name of the disease of COVID-19. And the symptoms in humans are associated with severe acute respiratory syndromes like high fever, dry cough, pneumonia, respiratory problem, lungs effusion, bronchitis, throat pain, multiple organ failure, and other symptoms like loss of taste, smell, occasional diarrhea. So it's become a global pandemic because till yesterday, SARS-CoV-2, it killed 8,98,000 people around the world, and 27.4 million people around the world get infected. In India, 4.28 million cases are there, out of which 72,775 deaths are there. So it's become the great threat. The problem is there is no specific vaccine or chemotherapeutics developed till date. So if there is no vaccine, no specific chemotherapeutics, then what will happen? It's a big query for our existence. How long does it take for discovery of specific anti-COVID-19 medicine? So, you know, for experimental design of a single molecule, it involves a series of reactions from the starting compound to get the final product. When we get the final product, structure elucidation is there by various processes, followed by biological activity evaluation by in vitro, in vivo studies, then structure activity relationship. So this long process, it takes a series of reactions which consumes a lot of money, lot of labor, and lot of time. Now, you see, drug discovery is a long journey and big challenges. Initially, 5,000 to 10,000 molecules are screened, which are called as hit compounds. That is called hit collection or hit generation. Then hit screening is there. For example, from 10,000, we have screened 100 hit molecules and synthesized. Then preclinical in vitro testing, followed by phase 1, phase 2, phase 3, phase 4 clinical trials. So, for preclinical testing, we tested 100 molecules. Then when it comes phase 1, it takes minimum 5 years. And around 10 molecules are tested in phase 1, followed by phase 2, which takes 7 years. 7 to 8 molecules are tested. In phase 3, it takes 10 years. And around 3 to 4 molecules are tested. When it launched into the market, means post-marketing surveillance in phase 4, only 1 to 2 molecules discovered, and it takes 14 years minimum. And it consumes 1,000 million US dollar. So, to find out 1 to 2 drug molecules successfully, it takes minimum 14 to 15 years with consumption of 1,000 million US dollar. So now, how shall we expect the vaccine or small chemotherapeutics, specific chemotherapeutics to combat the COVID-19? Because this drug discovery process, it's a long journey. So now query about our existence. Shall we live anymore from this infection? Or how can we protect the lives from the dreadful attack of COVID-19? Yes, we can live. How can we protect? By drug repurposing. What is called, what is it? It is called, what does it mean? Drug repurposing? Repurposing means
Sorry for the
existing
interruption, sir. Sorry for the interruption.
Sorry?
Excuse me, dear participant, Suhanji, Chauhanji, please, don't present your screen. Chauhanji,
Hello?
please don't present your screen. Yeah, there was interruption. No problem, sir. Just please continue.
Hello, sir.
Yeah, continue, please.
Okay. So, shall we live anymore? Yes, we can live. But how? How can we protect ourselves from the dreadful attack of COVID-19? By
so screen is not visible. Screen is not visible.
Sorry? Hello?
Visible, sir. Screen is not visible.
Hello?
Sir, your screen is not visible.
Okay.
No. Your
Hello?
slides are not visible, sir. Yeah. Now it is okay, sir. Now it's okay.
Hello? Hello?
Proceed, sir.
Okay.
Now it is okay. Proceed, please. Yeah.
Okay, so shall we live anymore? Or how can we protect ourselves from the dreadful attack of COVID-19? Yes, we can live. How? By drug repurposing. Now the question is, what is drug repurposing? So it's a method by which existing drugs, which is known for the treatment of one disease, that could be used or reused for the treatment of another unknown disease. That is called drug repurposing. Now, doctors have been treating the COVID-19 patient by existing antivirals such as remdesivir, antimalarials such as hydroxychloroquine, antibacterial such as azithromycin, antiparasites such as ivermectin, immunosuppressant or anti-inflammatory medicines such as prednisolone, natural immunostimulatory agents which are shown to have potent anti-COVID-19 activities. But what are the biochemical mechanism actions of this disease? We don't know till now. To get the biochemical mechanism of action of these repurposed drugs, we must have to study or explore the SARS-CoV-2 target. I mean to say different targets or enzymes which are associated for the disease virulence. You see, This is spike protein, which is very important target. It's a rod-shaped structure. This SARS-CoV-2 spike protein, it attaches to the human receptor ACE2, angiotensin converting enzyme 2, which is present in pneumocytes, I mean to say respiratory tracts, lungs, as well as in the gastrointestinal tracts. So this spike protein of the coronavirus, it can attach to ACE2 receptor of the host human cells, followed by fusion of the receptor and entry, which is done by heme-agglutinin esterase. So this spike protein, it attach with the nucleocapsid core. Another important is envelope protein. This envelope protein is responsible for pathogenesis and egress of the disease. Another important target, that is nucleocapsid core. This nucleocapsid core, it can protect the genetic material, DNA, and another important target is SARS-CoV-2 protease, main protease, and RDRP, RNA-dependent RNA polymerase. So this SARS-CoV-2 main protease responsible for replication of the viral genome. So therefore, first, most important target is spike protein that should be explored. You know, this is a spike protein, and in this spike protein, you see, this one is one homomonomer, this one is another homomonomer, and these two homomonomer, it attaches to the nucleocapsid core. And so this system, this is called homotrimeric structure. This system is called homotrimeric structure, and these two homomer, it attach to this ACE2 host receptor. Now, is there any inhibitor that can break down the cluster of this homotrimeric structure? This is one question. Another question is there any inhibitor that can inhibit the binding of homotrimer with the ACE2 receptor of the human? Yes, our scientists around the world, they have been trying to find out. Now you see, it was found that some natural compounds shows very good activity for the inhibition of spike protein of SARS-CoV-2. For example, curcumin, curcuma longa, nimbin, which is obtained from neem, uidaferin A, pipervarin, which is obtained from black pepper, thebain, berberin, andrographolide, imodin, tetraogaloyl beta-D-glucose, and luteolin. So these are the some important natural constituents which are shown as potential inhibitory activities against this spike protein of coronavirus diseases. Now, many vaccines are under the pipeline. Till September, there are 321 vaccines which are being going on. Out of 321, 33 vaccines are under phase 1 to phase 2 clinical trials. Out of 33, 6 vaccines came into phase 3 clinical trials. But no single vaccine till today has been successfully launched into the market. But what is the main difficulty? I told you, the main difficulty is to find out the volunteers with different age groups from multi-diverse countries. This is the main difficulty nowadays. Another difficulty is mutation of the spike protein. This is another difficulty. Now, I would like to present some update of COVID-19 vaccines which are under phase 3 clinical trials. The candidate, this is the vaccine name, AZ21222 by Oxford University and AstraZeneca. Next one is unnamed by Sinopharm Beijing, Wuhan Institute of Biological Products. Next one is CoronaVac by Sinovac, Institute of Butantan. Next one is BNT162A1B1B2C2 by Biotech, Fosun Pharma Pfizer. Next one is mRNA 1273 by Moderna. And next one is for India, Covaxin. And it works Bharat Biotech in collaboration with ICMR, NIP, National Institute of Virology. So these are the 6 vaccines which are being under phase 3 clinical trials. Now, so this is about the vaccine which can target this spike protein. Another most important target of the SARS-CoV-2 is main protease. So the coronavirus can attach the host target and causes fusion through heme-agglutinin esterase followed by the viral replication by SARS-CoV-2 main protease. Therefore, if we can stop or inhibit the main protease, then somehow the coronavirus disease can be inhibited, because SARS-CoV-2 main protease, it regulates the replication and transcription of viral RNA. These proteins are reported to inhibit the host cell, CDK4 and apoptosis. Therefore, if host cell CDK4 and apoptosis lost, then loss of our immunity organ. So anyhow, we must have to stop this main protease. How? It was found that existing antivirals and antimalarial chemotherapeutics can cause inhibition of COVID-19 by drug repurposing approach. That's why you see these are the some drug repurposed antivirals: remdesivir, oseltamivir, ganciclovir, ritonavir, lopinavir, favipiravir, sofosbuvir, ASC09, umifenovir, penciclovir, nitazoxanide, nafamostat, antimalarials chloroquine, hydroxychloroquine, antiparasitic ivermectin, antibacterials azithromycin, immunosuppressant anti-inflammatory drugs such as dexamethasone and methylprednisolone, and some herbal therapy I have told you. But exact mechanism of action of this disease are not found yet. So that is why it is our target in the present study to predict the binding affinity of this existing antiviral and antimalarials, to predict the mode of binding of these drugs towards SARS-CoV-2 protease enzyme. Now, you see, in my research, I have collected 34 drugs, antiviral drugs and antimalarial drugs, and some miscellaneous compounds. 11 compounds. Now the question is, so what is the basis? You know, out of 34 drugs, 11 drugs have activity against HIV-1 protease. 12 to 25 drugs have activity against hepatitis C virus protease. Now, why I have selected? Because protease is common for HIV-1 as well as hepatitis C virus. So, protease is also common in COVID-19. That's why protease inhibitor of HIV-1 and protease inhibitor of existing protease inhibitor of HIV-1 and existing protease inhibitor of hepatitis C virus may inhibit COVID-19 SARS-CoV-2 protease. That's why this is the hypothesis of my study. And some other antivirals like umifenovir, which is anti-inhibitor, oseltamivir, which is sialidase inhibitor for influenza, remdesivir, which is given for Ebola and Marburg virus, favipiravir, which is influenza RDRP inhibitor. These are also given for clinical treatment to inhibit the COVID-19. Then followed by some antimalarials such as chloroquine, hydroxychloroquine, and some other drugs such as ribavirin, penciclovir, nitazoxanide. Ribavirin is RNA synthesis inhibitor, penciclovir, herpes virus DNA polymerase inhibitor, nitazoxanide It is a commercial anti-protozoal agent. Now, these 34 compounds, these are the structures you see. Antiretroviral HIV-1 protease such as amprenavir, atazanavir, darunavir, fosamprenavir, indinavir, lopinavir, nelfinavir, ritonavir, saquinavir, pipranavir, TMC 310911. These 11 HIV-1 protease inhibitors. And hepatitis C viral protease inhibitors such as asunaprevir, boceprevir, siloprevir, danoprevir, faldaprevir, grajoprevir, glicaprevir, narlaprevir, paridaprevir, cymaprevir, sofosbuvir, sovaprevir, telaprevir, voxilaprevir. So these 12 to 25 serial number drugs, these are specific inhibitors of hepatitis C viral protease, and these are existing chemotherapeutics given for the treatment of hepatitis protease inhibition. And from serial number 26 to 34, you see umefenovir, oseltamivir, remdesivir, favipiravir, chloroquine, hydroxychloroquine, ribavirin, penciclovir, nitazoxanide. So these 34 compounds which are present in my biological activity data set. Now, in the process, what I did, these 34 compounds, chemical structure was drawn by using ChemDraw, then energetically minimized to get the most stable conformation of these compounds. X-ray crystallographic structure of SARS-CoV-2 main protease has been downloaded from Protein Data Bank. PDB ID code is IBAR7J. Then these compounds, these drugs, 34 compounds, have been docked into the active binding cavity of SARS-CoV-2 main protease target. By considering the co-crystal, name of the co-crystal is N25 fluoranyl 1H indole 3 ethyl ethanamide. It has been co-crystallized with this target, and this co-crystal has been taken as a reference compound to assign the binding cavity of my compound of interest, means 34 drugs, which I have shown as a ligand. Now results, you see, first binding activity, active binding cavity of SARS-CoV-2 main protease, it was analyzed, and it was seen that the existing co-crystal, this ethanamide, this compound can produce the important amino acid interactions with methionine 49, histidine 41, histidine 164, methionine 165, glutamine 166, and aspartic acid 187. So these 6 amino acids are very, very crucial for active binding of SARS-CoV-2 main protease. Now, you see, this is the docking results, and in these docking results, you see, out of 34 compounds, these 12 compounds such as amprenavir, darunavir, sofosbuvir, umefenovir, oseltamivir, remdesivir, favipiravir, chloroquine, hydroxychloroquine, ribavirin, penciclovir, and nitazoxanide. These 12 compounds, it shows binding or inhibition towards SARS-CoV-2 main protease enzyme. And in this table, I have shown the mode of interacting residues along with their dot score. And in this column, in the 4th column, some of the compounds in vitro activity were tested experimentally by some other authors. And this 4th column, I mean to say EC50, in vitro anti-SARS-CoV-2 main protease inhibitory activity, I have collected from the literature. Now, you see, this darunavir and umefenovir, these two compounds, darunavir, umefenovir, these two compounds, they are having mode of binding with this chloroquine, hydroxychloroquine, nitazoxanide. They are having mode of binding similarity. I mean to say, darunavir not yet tested, umefenovir not yet tested, chloroquine it has activity, and hydroxychloroquine the SARS-CoV-2 activity not yet tested. You see, these compounds, mode of binding, these are similarity, and their dog score -7.6, -7.8, -7.5, -7.5. I mean to say almost similar. Now, remdesivir, which is well-known, very well-known drugs for the treatment of COVID-19, now remdesivir injection has come into the market, and remdesivir, its mode of binding, is similar with this soposbuvir. Soposbuvir is hepatitis C viral protease inhibitor. So, in my study, this soposbuvir, hepatitis C viral protease inhibitors, it has been predicted as hello. Hello.
Yes, sir. Yeah, audible. Audible, sir.
Okay.
Yeah, full screen, please.
Okay. Now you see.
Okay, sir. Thank you. Okay, sir.
So this remdesivir, it has dog score of -8.5, and soposbuvir, it has dog score of -8.2, and mode of binding of soposbuvir remdesivir, so almost same. And the active binding residues of these highlighted compounds, they are similar with this methionine 49, histidine 41, histidine 164, methionine 165, glutamine 166, and ASP 187. So these drugs would be tested further for anti-SARS-CoV-2 main protease inhibitory activities. Now I have given these drugs mode of binding. You see, amprinavir, this is the best dose conformer pose of amprinavir COVID-19 main protease interaction, and this is darunavir compound. This is mode of binding of sofosbuvir. This one is remdesivir. This one is umefenobir. This one is chloroquine. This one is hydroxychloroquine, right side. This one is nitazoxanide. Now, how can I know? Because this is in silico docking study. This is prediction study. How can I know that this model, in silico model, is correct? So I would like to validate. I have validated this in silico docking model with the experimental results. In Wang et al., this author, they have tested these compounds: remdesivir, chloroquine, favipiravir, ribavirin, penicillin, and nitazoxanide. They have tested this compound against this strain, novel coronavirus strain of this SARS-CoV-2 main protease enzyme. And these are the activity results. You know, this is the activity results. And this is the dog score. Then what I did actually, you see, this is experimental activity. And this is the dog score. What I did, I make a correlation, quantitative correlation between these two parameters, EC50 and dog score. So this is the correlation result. And it was found that this correlation, it can produce R-square value of 0.899. If this R-square value is more than 0.5, statistically, it accepted as very good predictive in silico model. So my prediction has been validated with quantitative correlation between biological activity and dog score. And this is the equation: y is equal to minus 0.936x minus 7.631. This is the quantitative equation where x is equal to, x represents the anti-SARS-CoV-2 main protease inhibitory activity and y represents the dog score. Now, the conclusion is these 12 compounds, amprinavir, darunavir, sofosbuvir, umefenobir, oseltamivir, remdesivir, favipiravir, chloroquine, hydroxychloroquine, ribavirin, penicillin, nitazoxanide. These 12 compounds, these can be, I mean to say, very good, maybe very good potent compounds for the inhibition of SARS-CoV-2 enzyme. Now, this quantitative model which I developed, this quantitative model can be used for high throughput screening study of other existing drugs which are given for the treatment of, which are repurposed for the treatment of COVID-19 patients. Because I will show you some other thing also. And now, so my conclusion is, so through my prediction docking model, we have predicted these 12 compounds. Some of the compounds, biological activities there, like nitazoxanide, this chloroquine, remdesivir, and apart from that, other compounds which present in my prediction, these compounds could be tested for further anti-SARS-CoV-2 inhibitory activities. And this docking model, quantitative model, would be used as a template for high throughput screening of other compounds activity against anti-SARS-CoV-2 main protease enzyme. And from this lecture, I would like to give once more and repeated awareness to the society for maintaining the social distance, for mask use, sanitizer use, and intake of healthy food to maintain the health. And this original research has been published in Combinatorial Chemistry and High Throughput Screening very recently. And this work is in collaboration with Professor Midula Saxena, Amity University, and Professor Anil Kumar Saxena, Honorable Chairman of GPAR and formerly of Director Grade Scientist of CDRI CSIR. And some ideas which I would like to share with you for further collaboration. You know, new hopes come for SARS-CoV-2 treatment. It was found that in Nature Microbiology, very recently it is published, it was found that this LY6E, this is a gene of SARS-CoV-2. It can inhibit, means coronavirus gene 6Y LY6E. It can inhibit coronavirus entry by inhibiting spike protein, human ACE2 receptor complex, and membrane fusion. I mean to say, one gene of coronavirus, that is 6LY6E, that can inhibit the coronavirus entry into the human body. So somehow, if we can design and discover some novel chemotherapeutics that can activate the LY6E gene. So this is a very good concept, and this is a very good idea. I'm not doing the bioinformatics work, so this is open for collaboration. And you see, it was found that cefixime, cefodoxime, ofloxacin, sparfloxacin, and doxycycline, this has been given for the treatment of COVID-19 patient. What is their mechanism of action? What is the mechanism of action of these drugs? It was not yet done till now. So this is another idea. Another important idea is, quantum sensing mechanism. I mean to say, the virus, this type of virus, they can maintain the quantum sensing mechanism. Quantum sensing means this is a methodology of intercommunication between each other. I mean to say, so when chemotherapeutics are given to kill the coronavirus or virus, then one virus, it pass the message to the adjacent virus that chemotherapeutic drug has administered into the human body, so you please be careful. So this type of communication is there between the virus, which is called as quantum sensing mechanism. So my idea is whether anti-quantum sensing drugs may be helpful along with this existed repurposed anti-COVID-19 chemotherapeutics. So these are the some 3 ideas which I would like to share to everybody for open collaboration. Finally, I would like to acknowledge IIT Roorkee, Water for Welfare and Uttarakhand Initiative for the project fund, from which we have purchased the workstation. In our institute, we have purchased this from the Singapore. It's a very powerful workstation for drug design work, because in our institute, one drug design laboratory has been established by the management, and I am looking after this drug design laboratory for the work, and we have many collaborative works. And I am fortunate to achieve training from Dr. Anil Kumar Saxena, who is Honorable Chairman of this institute and formerly of Director Grade Scientist CDRI CSIR. He is my academic Guruji. He has trained me a lot. I am grateful to my parent teacher, Professor Kunal Rai, Jadavpur University, Kolkata, and my mentor, Dr. Monish Sivachi, formerly from IICB CSIR, Kolkata. And I am sincerely thankful to my collaborators, Professor Dr. Nidula Saxena. She is my Guru Ma from Amity University, Lucknow, Uttar Pradesh. Dr. Asmita Samadhar from University of Kolkata, my elder sister, West Bengal, she is my collaborator, and Mr. Hare Krishna Rai, Nehru College of Pharmacy, Guntur. And I am sincerely thankful to my potential students like Mohit, Ankita, this Ruby, Salman, and Amit Kumar. This Amit Kumar, he is the fellow of INSA, and these potential candidates I have, expert hands I have. Because of them, I published many articles in impact factor journals, and my students are very much active. And I am happy to share you that BPharm final year project students and M-Pharma project students, they did a lot of work and they published their project work in impact factor journal. Like you see, this serial number 2, Mohit Kumar, this combinatorial chemistry high throughput screening, impact factor 1.503, and many others work of other students published in impact factor journal like Springer, Bentham Science, Wiley. And finally, I salute my mother India. I am proud to be Indian because we have a stable immunity. Thank you all. Now the lecture is open for discussion. I shall be very happy if you raise any question or ask me any question or give constructive suggestions. Thank you.
Thanks a lot. Thanks a lot, Dr. Suresh sir. Delivered the lecture on repositioning of chemotherapeutics to combat COVID-19. The delivery was very interesting and informative session. Yes, sir. Thanks a lot. Very interesting and informative session.
Yeah, thank you very much, sir. Thank you.
On current strategy of COVID-19 pandemic. So your presentation explained about the current challenges in discovering the new drugs, and you have taken a very great initiative for the molecular docking studies, which explained very clearly.
Yeah.
And with the evidence of your excellent research work, I wish you all the best for the future research.
Yeah. Thank you, sir.
Yeah. So now, sir. Welcome, sir. And now the time for discussion for the participants, please.
Good afternoon, sir. Can you hear?
Yes, madam.
I am Dr. Subin Mary Sakriya from Amrita School of Pharmacy, Kochi.
Madam, just a minute, please. Just a minute. Please, other participant, please mute your mic. Other participant, please mute your mic. Yeah, you can proceed, madam. Thank you.
Yes, sir. Sir, your session was very interesting. It was very relevant to this present situation. Very relevant to this present scenario. Sir, I have one question. This combination of acyclovir and azithromycin was proved to be very effective for COVID-19 patients.
Yeah.
Your opinion on that, whether you have done any repurposing studies on that combination, protease inhibitor acyclovir and azithromycin.
Hello.
Can you hear me?
Yes, please, ma'am. Please continue.
Yeah, sir. Actually, the combination of azithromycin and protease inhibitor acyclovir was very effective. It was proven to be very effective for COVID-19.
Yeah.
Repurposing. Yeah. So, what is your opinion on that?
I understood your point. Ma'am, hello.
Yeah, sir.
I understood your point. I can explain it well. So, this is what you said. I appreciate your thinking because this is now I am working on azithromycin in silico developing, and the manuscript is under formulation, and I am sharing you. It will be submitted very soon that azithromycin has potential inhibitory activity against main protease. This result came into my study.
Okay, sir.
And another important point is there because as it is not published, I don't want to share. Azithromycin is well known for anti-querum sensing activity. That I told you. Idea number 3. In this slide, you see the idea number 3, which I would like to share. Do anti-querum sensing drugs helpful along with repurposed anti-COVID-19 chemotherapeutics? Yes, it would be helpful because if you study the querum sensing inhibitors, you can find that azithromycin is a very good drug for inhibition of querum sensing mechanism of the virus and bacteria. So this would be another mechanism of action that is also under my study. So these are the two important observations that azithromycin is a very good drug for COVID-19 patient to treat the COVID-19 patient because it has predicted activity against SARS-CoV-2 main protease as well as anti-querum sensing agents. Thank you very much, ma'am. Thank you for your answer.
Because it has proven, it is very proven that this combination is very effective, especially in Middle East countries. This combination is giving. right now. That is why I asked. Okay, sir. Thank you. Thank you very much.
Thank you, ma'am. Thank you.
Any more questions from the participant side? Dear participants, any more questions? The time for discussion. Sisi sir.
Yes.
Yes, yeah, yeah. The work was very this one. Just I want to ask a practical question. So now
Yes.
the situation. How long it will take for this one? So for a drug to come out in the market. So for in this present situation, worldwide, yeah, different researchers are going on. You have collected a very good,
Yeah.
we have collected a very good information from the different parts of the pharmaceutical companies, what they are carrying out the drugs discovery. So how long it will take it comes into the market?
1 minute, sir. Actually, it's a big question, you know. I have told you. You see, this is the general drug discovery process. It takes 14 years to discover a medicine. So, in that pandemic situation, if the vaccine will not be developed, then lives, more and more lives, uncountable lives will be lost. And we are going to lose the national integration of each country. So under this chaotic situation, scientists from all over the world are doing collaborative work, and they would like to launch the medicine as soon as possible. Remdesivir has already come into the market as injectable formulation, and favipiravir, two companies, favipiravir, small molecule tablet, it has to be launched very soon, expected within 2 to 3 months. Next 2 months, favipiravir will be coming into the market. And favipiravir success is 74%. The study shows favipiravir success 74%. We must have to maintain the social distancing. We must have to use mask. We must have to use the sanitizer for protect ourselves from the attack of these dreadful viruses. And we must have to unite together to combat COVID-19, because this is a very contagious and dangerous disease. You know, Earth is a planet and we are the lives. We must have to save our Earth, to save our life. So we must have to unite together, and we must have to do the collaborative work to fight against this disease.
Oh, thank you, sir. Thanks for your explanation. Wonderful explanation. There is another question from the participant side. Should I read out?
Please tell me.
Yeah. Mr. Sarfaraz Ahmed has raised a question. Mutations are occurring in a coronavirus. So what effect will the mutations have an outcome of the corona vaccine?
Yeah, this is actually.
Yeah, mutation, that is a big issue now, sir. Every region it is happening. There was a lot of changes. That's what the discovery is there. So
Yeah.
how it is happening? What was the outcome?
Yeah, actually mutations are going on. That is why this is a big problem for the development of particular drugs. But scientist Orishon, I have not shown here it because the manuscript is under submitted now in current current topics in medicinal chemistry. One scientist there, scientist Orishon, he has explained that the mutation does not increase the severity of the disease. It increases the contagious property of the disease. Somehow he has explained, but I don't know, because this mutation is a big problem for the development of the vaccine. Because so far as I have read and I have knowledge, I have very poor, very limited knowledge on the vaccine development. So mutation is a big problem because if mutation vaccine is developed on the basis of some strains, and if it is mutated, then what will happen? Maybe it will not work, or it will work in some country fellows. So this is the problem. So that's why for the discovery of vaccination, the main difficulty is to find out the volunteers with different age group and multi-diverse countries. In India, we have more than 130 crore colleagues, citizens. So around the world, many populations are there, a big populations are there. So how unique vaccination will be there, development, one vaccine which could be unique or could be fruitful for all over the world, that is a big question. That's why development of vaccine, it will take a lot of time. I think so, it will take a lot of time. Because
thank you.
For this phase 1, phase 2, phase 3, phase 4, this trial or testing, it will take time because lacking of volunteers with different age groups from different countries. But our scientists have been doing the collaborative work, so we expect that it will be coming, it will be launching to the market very soon. And already I have shown you, these are the 6 vaccines which are under pipeline in phase 3 clinical trials.
Thank you, sir. And another question is there. Kindly, possible, just give, please see through the chat box, sir. Chat box. It will be very clear there.
1 minute.
There is one question. Yeah.
Yes.
Please check. Please check in the chat box. Mr. Mayank Kumar. Mr. Mayank Kumar Kede.
So Mr. Mayank Kumar has written when we can expect this pandemic will end. So no, we cannot say anything about this. When this pandemic situation will be ended, we cannot say. But the thing is, the lives, human lives, and these natural lives, I mean to say plants life on the earth, this is for million years. This is existed for millions of millions years. But virus, the lifespan of virus, it's not more than the lifespan of the human, and human brain is also very much potent than the viral brain. So we as a human must discover the medicine and we must save us. But we cannot say when this pandemic will be ended. We cannot say because this is a very contagious disease, very contagious disease. And this transmission, contagious pathogenicity or infectious transmission, it will be carried out in a chain order. And if you see before, 100 years before this Spanish flu, it took 3 years to recover, but that time population was not so large, and now population is so high. So this disease transmission, it is going on in combination permutation disorder, permutation combination disorder, permutation combination statistical approach. This pathogenicity or contagious property of this disease has been spreading. So we cannot say when this pandemic problem will be ended. We can expect the medicine, particular medicine, and if medicine, when medicine will be discovered, then that infected patient will be treated. But like like influenza or other this viral fever, so we must have to save ourselves from this problem. And I think it will be for the next 5 years, 5 to 6 years, I think so. And it will be the part of life. But we must have to be very careful. We must have to drink healthy food. We must have to eat the healthy food. We must have to drink healthy drinks, and we must have to avoid our hi-fi lifestyle, like this together drinking alcohol, which can affect the liver, kidney, and intake of the spicy food like non-vegetables, chicken, mutton, biryani, or junk food, or this party attending party, and this unhealthy lifestyle. This must be avoided for the next 5 to 6 years to combat against these diseases. But one expectation is there that herd immunity is going to be formed. That's why rate of the recovery, percentage of recovery has been increasing in India. And death rate is going to decrease. Rate of infection has been spreading from 30,000, 40,000, now more than 80,000. But rate of recovery is also increasing, and death rate is also decreasing because of development of the herd immunity. And that is very nice. If we maintain the healthy drink, healthy food, our immunity will be increased. We must have to fight these diseases. And the senior citizen, more than 60 years age, fellow, and the baby, they should also be, we must have to protect them.
Wow, a very good explanation, sir. So for time being, this is the only solution to increase the immunity.
I think this is the only solution, and the important thing is that we must have to avoid this unhealthy lifestyle. What I told you, unhealthy lifestyle, regular intake of breakfast, lunch, dinner, very much important. Regular intake of vegetables, fresh vegetables, this fibrous, protein, carbohydrate. Equally present, fresh vegetables we must have to intake, and we must have to avoid this. I mean to say, much amount of non-vegetables like chicken, mutton, biryani. The non-vegetable who are taking the non-vegetables, they take and they are taking. But apart from non-vegetables, we must have to dependent on the natural vegetables, which can increase our immunity. Because there is no substitution for mother. In this world, there is no substitution for mother, and there is no substitution for natural medicine and natural vegetables. I believe that, and everybody believes that. And nature is our mother, and our mother nature can protect us only.
Yeah, great, sir. And there is another question. Kajalban Patel has raised a question. Some cases are reported with the reoccurrence of COVID-19. Can you please explain about?
Yeah, I can say this. One of my very good friend, who is my jigridos, like very close friend. He is a doctor from Kolkata Medical College, and he was doing duty in Kolkata, Beleghata, this hospital. To treat the COVID-19 patient. I have regular discussion with them and get some potential knowledge from him, which helped me to carry out this research work. And the thing is, when a COVID-19 patient is going to recover, he has been dispatched from the hospital with certificate that yes, everything is okay. But the patient is not okay for everything, because already I told you, you see, Already I told you in this slide, you see this SARS-CoV-2 main protease, it can attack the immunity, loss of immunity. It can inhibit the host cell CDK4 and apoptosis and loss of the immunity. So when patient recover, to regain the immunity, it takes minimum 2 to 3 months, practical. Because I discussed COVID-19 patient also. The recovered patient I discussed, face-to-face discussed. So he is physically, the patient is physically very ill. And to at least 2 to 3 months, it takes to regain the immunity. So under this condition, when immunity below the permission, the patient must be very careful to expose to the great community. I mean to say to expose to the environment. So when, okay, one patient recover and within 1 month, if he thinks that, okay, I must have to go here and there, and I must have to meet with many pupils like or without social distancing. No, it's wrong. He or that patient who just recovered, he or she might have attacked not only this coronavirus, but also other viral and bacterial problem. So that is why this problem reappearing. But after 3 months, when healthy food maintaining, healthy drinks maintaining the social distance, after 3 months, the possibility of getting corona infection is least chances. This, as per the report of the doctors who are treating the COVID-19 patient. Because as a pharmacist, I would like to do something for the society. What can I do? So I can do some potential research work, and I can do some social awareness, and I can advise to my environment or colleagues who are associated with me that maintain this type of food and maintain this type of lifestyle. And if I say 100, if I inform to 100 colleagues, and if one colleague, he listen or he follow, then my aim is successful as a pharmacist. I think so. Because I would like to always motivate myself to devote myself to do some constructive work in my society. And this has been taught by my institution, where Dr. A.K. Saxena is the chairman, very eminent scientist and my Guruji. And he always told to everybody, each and every faculty, that, okay, you do constructive work and always involve yourself through research. As an academic teacher, this is not only my satisfaction if I do carry out research work, do some constructive work for the society. So I am satisfied by my own. So other researchers, other pharmacy faculties who are in meeting and you are listening my words, you also motivate to your colleagues and please you gain knowledge under this pandemic situation and protect your colleagues' life by social awareness. At least if you gain some knowledge, you can protect your family, you can protect some colleagues' life also. This much of integration we should have, because coronavirus kills the national integration of each and every country. I am confined in Uttarakhand, I am unable to move Delhi, I am unable to move West Bengal, I am unable to move other parts of my mother's land, I am unable to move. So this type of integration is going to be lost around the world. It should not be. We must have to unite it, and we must have to cautious to protect our family members, to protect my colleagues' life also.
Very good explanation, sir. There is, I think, maybe the last question. One person, one person has raised a question. Jansi M. If one person is already suffered with flu, the chances of getting corona?
Sorry, sir. Can you please repeat?
Yeah, it was there in the chat box. If Jansi M has raised a question that one person already suffering with the flu, the chances of getting COVID?
Yes. If one person is suffering from flu, is there any chances for COVID?
Yeah, that was the question.
Yes, it is. Because when a patient is suffering from flu, I can explain. When a patient is suffering from flu, viral flu, then fever is there. And if fever is there, means something is wrong. Immunity, I mean to say body's immunity system is fighting against that flu virus. So immunity loss and body power will be lost. Under this condition, if that patient is not being isolated, then he or she, that patient will get chances of coronavirus infection. Of course, it will be in touch with coronavirus, of course. I mean to say, if that patient is being isolated, there is being no chances. If that patient is exposed to the market or public or other, I mean to say, environment where viral zone is there in touch, so that patient might have chances of getting infection, coronavirus infection.
So now it is completely asymptomatic, no? So no, throughout the globe, so how we can identify a person is infected or not?
Yeah, so that is why the government instruct, advise to all community that maintain the social distancing. This is the only solution, because if anybody is well immunized system, biological system, then asymptomatic will be there. So when he exposed to the environment where a fellow, senior citizen fellow, or a fellow having cardiac disease or liver disease or chronic kidney disease, that fellow, he will be getting infected. So that's why social distance is only solution, social distance. And I advise, when there will be rain, because coronavirus is floating on the air, when rain will be there, then people must be within the four wall, within the roof. They should not be drenched by the rainwater. So the people should not be in contact, in close contact with other people, because we cannot predict whether in front of the people who is sitting in front of me, whether he or she is having this asymptomatic problem. That's why this social distance is only solution right now to protect ourselves. But in this condition, shall we confine within the four wall? No, because we have to earn the money, we have to do the work, we have to do our regular activities. Otherwise, how shall we earn the money? How shall we maintain the family? How shall we maintain us? If you think a lot, if you confine within the four wall, you will be mental patient. And then it's a big problem. Coronavirus create other problems, social problem also, like quarrel between the husband, wife, quarrel in the family, fighting in the family members, mental problem, like that. This problem also arises. So to minimize this problem, other problem, we must have to be active in our daily life with maintaining the precautions like social distancing, mask, sanitizers, and must avoid the party with where accumulation of some other public, like that in public, must have to avoid the party, attending the party or invitations like that, where many gatherings are there.
Yes, sir. Another question from Mr. Vijay Bhaskar.
Yes.
Some nations are having strong belief on herd immunity, and they are not implementing strong social distancing restrictions. How about your view in the herd immunity related to COVID?
Sir, please repeat it again, please, sir.
Sure. Some nations are having a strong belief on herd immunity.
Yes.
And they are not implementing strong social distancing restrictions.
Yes.
How about your view in their herd immunity related to COVID-19?
Yeah. You see, this herd immunity concept, it was announced at the beginning, in the beginning of March, February, March. So, because of when this announcement was there, you can see in the internet. I have listened the news in television. This when this herd immunity concept was announced, like Germany, Italy, and this USA also, these people, young people, they go outside and they get the corona infection. When they come back into the home with the infection, this fellow who has lacking of immunity, like babies or some other, that senior citizen fellows, they get infected and they lost their life. So herd immunity concept, this is absolutely valid because it is well proved, experimentally proved. But coronavirus, SARS-CoV-2, it has been pandemic, it has been erupted in late December, in the end of 2019. And when this virus erupted, that time it has a big power to kill the community, to kill the lives. So that time herd immunity concept fell. Herd immunity gradually developed. We must have to keep in mind, herd immunity gradually developed. Now coronavirus mutations are there. Mutations, many mutations are there. So upon mutation and charging of chemotherapeutics, the virulence, I mean to say, power of the dangerous activity to kill the lives, this virulence will be losing day by day, and herd immunity will be gaining day by day. When virulence will be decreasing, then herd immunity will be increasing. So I am a healthy fellow, when well immunized fellow, when I expose to the environment where coronavirus is there, I will get that infection. I mean, anybody can get that infection. But my body is well immunized and fighting and kill the coronavirus. And this fighting develops the antibody to develop this herd immunity. But when the pandemic disease began, that time herd immunity concept fell. Now this is gradually developing, but still we should not be in together. I mean to say, we should not be in together doing the party attending, accumulation of the social people in a party. It should not be, because we don't know who is infected with the dangerous strain. We don't know at all. But as an Indian, I can say we are having the stable immunity, and we must maintain these government instructions or advices, and we must fight. We should fight to complete recover from this problem. We must. Hello? Hello?
Yeah, sir. Yes, sir. Thank you very much. Yeah, very good explanation, sir. Very good explanation. Participants, any more discussions, any more doubts? You can text also.
Very good question. I have seen one question in the box.
There is an
sir, is there any severity change of the attack of coronavirus during winter session? This is a very good question. You know, in the part of Himalaya, like Uttarakhand, Darjeeling of West Bengal, like this Jammu Kashmir, this in the part of Himalaya, there is a big winter. I mean to say, the temperature goes almost 0 or less than 0, like that. So, but the thing is that how to say, in Himalayan region where hill is there, population density is less. So population density is a big factor, because coronavirus carrier is very important. Try to understand what I'm explaining. Coronavirus carrier is very important. In Kolkata, population density is very high. In Hyderabad, Chennai, In New Delhi, population density is very high, so number of coronavirus carrier will be more. In that region, problem will be more. But in hill area, the problem will be less because population density is less, and also in hill area, oxygen pressure is more due to tree, natural tree. So when there will be lot of oxygen pressure because of natural beauty, automatically pollution is less. At the time of winter, we have seen in the previous year that at the time of winter, when winter just going to summer dissolve, winter just going to start it, people suffer from common cold, coughing, common cold. And when winter is going to finish, that time also. So these two times people suffer. In between also people suffer. But the thing is, at the time of winter, when there will be cold and coughing, it will be a problem. Problem for everybody, not only for baby, not only for senior citizen, problem for others also. Because in India, the pattern is infection has been spreading by permutation combination approach. And AIMS director of AIMS New Delhi, he has announced in the television that from a mathematical model has been developed, and that mathematical model, it was told that in the month of January, December, January, in India, there will be infection per day more than 1,50,000 minimum. So we must have to be careful, because now it is September, October, November, December, 3 more months are there. From December, from 10th of December, the winter will be more and more till February. So this time we must have to be very much careful by wearing the winter clothes, obviously gloves, socks, and we must have to maintain our lifestyle. So during the winter, we can expect some problem, more problem. It has already been announced by Government of India, Director of AIMS.
So there is 2 more questions in the chat box.
What is the question, sir? Hello?
Sir, there are 2 more questions in the chat box.
Who has raised Ruby Jadhav?
No, Mayank Kumar Kade.
Acha, Mayank.
Next is
what is your opinion on existence and origin of the virus? May theories around world claiming it's lab made and not organized naturally. What is your opinion on this? Many theory also claims it's naturally originated. It's a topic of debate. Okay, just 1 minute, please. I would like to show you one model. Hello, can you see this model?
Yeah. We are seeing. Yeah, coming.
It's coming.
No,
Okay,
not yet.
it's coming now.
No, sir.
Is it coming?
Coming, sir.
Can you see this model? Hello?
Yeah, it is blank, but nothing is showing, sir.
Nothing is there.
Yeah.
Now can you see? Can you see? Hello? Can you see the model? No.
Screen is blank.
Why?
Yeah. Picture is not there. No picture. No pictures. Only blank.
This one?
No, sir. You give the present screen and the entire screen and select the entire screen.
Select entire screen. This one. I have selected.
Yeah. It's showing only the blank screen, sir. Only black.
Okay. But this one is coming or not? No.
No, not yet, sir.
Why? What is the problem? Again now. Is it visible? Okay. I don't know, some problem is there, technical problem.
Yeah, coming, sir. Now it is coming.
It is coming now, model? Okay.
Yes.
You see, this COVID-19 infection, Mayank, you see, COVID-19 infection is there, and it can infect the animal, like cats, bats, and from infected animal, healthy persons are infected. This, in the box, some colors are there. It represents the gene, you know. So, from animal to healthy human to animal, there will be cross-species transmission in this case. And due to this cross-species transmission, this, what happened? I don't know. Some problem is there. Hello, can you hear?
Yeah. I'm hearing, sir.
So
Hearing. Voice is audible.
From infected animal to healthy human to infected animal, there will be cross-species transmission. Because of this cross-species transmission, highly
Screen is not.
resistant strain
Sir.
is produced.
Hello, sir.
Yes.
Screen is not visible.
Coming? Hello,
Yeah,
sir. Coming?
yeah, sir. Yeah.
Because of the cross-species transmission, there will be highly resistant strain. I mean to say combination of genetic combination of animal gene, combination of healthy gene in this highly resistant strain. That is very dangerous. So this highly resistant strain cause infection to the healthy individual and immediate multiple organ failure is there. So from this study, it was proved that it has come in the bats or cats. So it is animal, I mean to say cats or bats originated. But till now, it has not been proved whether it has been mutated in the laboratory. So we cannot claim anybody legally because it has not been proved in the document till now. But it is naturally, I mean to say cats or bats oriented. And I have told you at the beginning, it was found in 1930 in poultry chicken. So coronavirus is not the new one. It is very old thing. And this SARS-CoV-2, which is called as novel coronavirus, that is the novel strain or new strain, erupted in late December 2019. So whether it has been mutated in the laboratory or not, it has not been proved till now in the document. So we cannot claim and we cannot share single words on that. Okay.
Yes, sir. Thank you. There was a last question.
Yes.
Yeah. Ruby Yadav raised a question that patients suffering from COVID-19 are made to sit under sunlight for vitamin D. So during winters, this would be a difficult and main concern. What is your idea, sir?
Yeah, because from patient counseling, I talked to one patient, this COVID-19 who was having the COVID. She is a lady of 46 years old. She was having the coronavirus problem just in the 15 days, 25 days before, 1 month before, here only in my area. And we are in contact with that family. I had a telephonic discussion with that lady, and she is having a problem of vitamin D3. After recovering, she has been tested hemogram and tested the vitamin D3. After coronavirus, she is having the vitamin D3 problem. That's why she is having very weakness, body weakness. So vitamin D3, lacking of vitamin D3 is a big problem after corona infection. So how vitamin D3 obtained? We obtain vitamin D3 synthesis in the body by using the sunlight. So if time permits, the people, they can get sunlight in the corridor at their home, or maybe they have to maintain the time when other people not be there in the roof of their own home. So they can they can gain the sunlight, natural sunlight. There is no substitution for natural sunlight. We cannot substitute natural sunlight with the medicine. But if coronavirus created vitamin D3 problem is there, then that patient must have to take the medicine, vitamin D3 medicine, with consultation of doctor through proper channel. Because immediately after recovering from corona, sunlight does not repair that problem. That patient must have to take the vitamin D3 tablet or marketed formulation in consultation with the doctor. That is absolutely correct. But during, health is wealth. So we cannot purchase the health by using money. A person is billionaire, crorepati, crores of crores money, but his health is not good. That person is the saddest person in this world. If a person is well health, then he is the happiest person in this world. So to maintain the health, we must have to pay the time and we must have to enjoy the sunlight, which is natural source. And I told you, there is no substitution for natural and mother's affection. Okay, thank you. Please tell me if you have any more questions.
Thank you very much for your wonderful explanation, sir, for all the questions, as well as your patience for hearing and questions and answering very politely. Thank you very much for your valuable time spent with us. Thanks a lot, sir. Thank you very much, sir.
I would like to share one message to the participants. I would like to share one message to the participants. Hello?
Yeah, proceed, sir. Proceed.
Yeah,
proceed, sir.
I am working in the field of drug design, computational screening, computational drug discovery. Now, as this COVID-19 is a very contagious problem, so experimental BSL, means biosafety laboratory, at the top level BSL setup is very difficult. Government also motivate. If you see the government guideline, Government of India guideline, DBT, DST, ICMR guideline, government also motivate the young researchers like pharmacy faculty, biotech faculty, chemistry faculty, bioscience faculty, or other faculties who are associated with the research. Government motivate to do computational screening work. And now hackathon drug discovery, DDH drug discovery hackathon, Government of India, they have announced, and it is going on. In drug discovery hackathon website, there are a lot of ideas which have been shared by the government. So faculty or researchers, students, research scholars, M.Pharm scholars, B.Pharm final year scholars, they must they must see the website of DDH, and they can see a lot of ideas are there, and they can solve that ideas, and they can publish their own work, they can do their PhD, they can do their M.Pharma, everything. And now government motivate the computational drug discovery, means through drug repurposing. Existing drugs are there. You check whether these existing drugs are active against corona, because BSL setup is a big issue. It's time consuming and it's our protection also. We need a lot of measurement, lot of precautions, which is not possible. At the top level it is possible, but the pharmacy institution, the setup of BSL, it is very difficult. That's why you do the computational screening and you propose that molecule to ICMR. They are charging some money, and also CDRI, you can propose CDRI also. So they will be getting tested. So please, young researchers, you must do something for our country, because you do some drug design work, computational works. Don't think that computational works takes a lot of money. I mean, it is very expensive. No. Software is very expensive, it is true. Like Schrodinger, it is very expensive. But some softwares are there which are Google free. I can share with you some softwares. For molecular docking, Argus Lab 4.0.1, please note it down. Argus Lab, ARGUS, Argus Lab LAB 4.0.1, Argus Lab, this version is free, Google free. You can download it. You can download the protein from this PDB site, and you can do the molecular docking. You read natural compounds. You read that Government of India guidelines like WHO and our ICMR's guidelines. There are a lot of drugs which are repurposed. You take that drugs and you do the molecular docking, and you proposed through proper channel. I can share this message that through Google you download in your own laptop. You have 30,000, 40,000 rupees laptop you have, everybody you have. So you download it and you do the molecular docking. And if you want, I can I do always help to you. I can help to you. I am ready to get you help. My email ID is there with Professor Sudhahar, and you can call me anytime. You can take the advice, and I can teach you through Google Meet also how to do the this molecular docking study. I can help you. I am ready to help you, because you know, if I share money from my pocket, my money will be decreasing. But if I share knowledge from me, my knowledge will be increasing. So this is the only point, knowledge. When you share with other people, so your knowledge will be increased, you will be enriched, and other fellow, pharmacy fellow, who are your colleagues, he will also be enriched in this collaboration. So we can must protect ourselves from this attack. So please, pharmacy researchers, young faculties, don't stay at home. You do work, you do the computational screening. Many softwares are there which are Google accessed freely. You can download at your laptop and you can do the computational study. And I would like to share one words. In India, in 2017, our institute organized CMTPI, Computational Methods in Toxicology and Pharmacology, Internet Integrated Resources, this CMTPI conference in Goa. Our institute was the host institute. I was the organizing secretary, and Professor Anil Kumar Saxena, he was the chairman, because he is the honorable board member of a union, CMTPI body of European Union. India got the first chance in 2017, and our institution, they organized where more than 16 countries scientists came at Goa in India, first time. And what is the basis? What was the basis of that conference? The basis was how to access Google free software. So I am experienced with Google free softwares as well as commercial software. So I can always help you what are the different names, how to operate, how to access that softwares. So please, you do something for your own satisfaction, for your own enrichment of the knowledge, as well as to combat COVID-19. So this is my message to everybody. Thank you, sir. Thank you.
Yeah, thank you, sir, for a very wonderful session. And it was more interesting as well as very informative. Thank you very much, sir. Thank you very much. Yeah. Rajshri madam is there?
Yes, sir.
Yes, please conclude.
That was such a relevant and informative session, sir. Thank you, Dr. Shishinandi, sir, for such a session. I hope all the delegates got a very informative session. On behalf of Nehru College of Pharmacy, I extend my thanks to Dr. Shishinandi, sir. Thank you, sir.
Thank you. Thank you. Thank you.
And also, I thank all the delegates who participated for today's session also. I request everyone to join to the same link tomorrow at 9:30.
Thank you. Thank you. So today the session is finished, sir?
Today's session is over, sir.
Okay, so shall I quit?
Yes.
Okay.
Thanks a lot. Thank you. Thanks a lot.